Movement is so fundamental to human existence that we rarely pause to consider its mechanics. From the moment you woke this morning, reached for a glass of water, or even continued breathing as you drifted back towards sleep, your muscular system has been working. Quietly, continuously, and for the most part, without your conscious involvement. This is the biological machinery that transforms intention into action, that converts neural signals into physical reality. Over the next few hours, we'll explore exactly how this happens, from the molecular events inside muscle fibers to the coordinated contractions that allow you to do, well, anything at all. Muscle tissue is, at its core, a biological answer to a physical problem. How does an organism move itself, or move things within itself, without rigid mechanical parts or external motors? The solution evolution arrived at is contractile tissue, cells that can shorten themselves on command, pulling whatever they're attached to closer together. It's an elegant solution, really, though like most biological systems, it comes with considerable complexity beneath the surface. There are approximately 640 named skeletal muscles in the human body, though the exact count varies depending on how you're counting and which anatomist you ask. Some muscles that appear singular are actually composed of multiple distinct sections, while others that seem separate share origins or insertions. Regardless of the precise tally, these muscles make up roughly 40% of body weight in the average adult male and about 30% in the average adult female. This is a substantial investment of biological resources, which gives you some sense of how important movement is to survival. All muscle tissue, regardless of type, originates from the mesoderm, the middle of the three primary germ layers in the developing embryo. During the third week of development, the mesoderm begins to differentiate into various tissue types, and by the fourth week, muscle precursor cells called myoblasts begin to appear. These cells are committed to becoming muscle and nothing else, though they haven't yet developed the specialized structures that make muscle tissue functional. The process by which these cells transform into mature muscle fibers is called myogenesis, and it's a fascinating journey from generic precursor to highly specialized contractile cell. The defining characteristic of all muscle tissue is contractility. This isn't just the ability to change shape, which many cells can do. Contractility refers specifically to the ability to generate force while shortening. When a muscle cell contracts, it pulls on whatever it's connected to, whether that's a bone, another organ, or even another part of the same organ. This is accomplished through the coordinated action of specialized proteins arranged in precise geometric patterns within the cell, a mechanism we'll explore in considerable detail in later chapters. But contractility isn't the only notable property of muscle tissue. Muscle cells are also excitable, meaning they can respond to stimulation by generating electrical signals. This excitability allows muscles to communicate with the nervous system and to propagate signals across their surface, coordinating contraction throughout the cell. Muscle tissue also demonstrates extensibility, the ability to stretch beyond its resting length without being damaged. This is important because muscles are often stretched by the action of opposing muscles or external forces, and they need to accommodate this stretching without tearing. Finally, muscle tissue exhibits elasticity, the ability to return to its original length after being stretched. This property helps muscles maintain tension and position even when external forces are applied. There are three distinct types of muscle tissue in the human body: skeletal muscle, cardiac muscle, and smooth muscle. These three types share the fundamental property of contractility, but differ substantially in their structure, function, location, and control mechanisms. Understanding these differences is essential to understanding how the muscular system as a whole operates. Skeletal muscle, as the name suggests, is the muscle attached to the skeleton. This is the tissue responsible for voluntary movement, for locomotion, for posture, for facial expressions, and for the manipulation of objects. Skeletal muscle cells are long, cylindrical, and multinucleated, sometimes containing hundreds of nuclei in a single cell. This multinucleated state results from the fusion of many myoblasts during development, a process that creates cells that can extend from one end of a muscle to the other, sometimes spanning lengths of 30 centimeters or more. Under the microscope, skeletal muscle appears striated, marked by alternating light and dark bands that reflect the highly organized arrangement of contractile proteins within the cell. The control of skeletal muscle is voluntary, at least in principle. You can decide to lift your arm, and your arm lifts. You can choose to walk forward, and your legs carry you forward. This voluntary control is mediated by the somatic nervous system, which sends conscious commands from the motor cortex of the brain down through the spinal cord to motor neurons that innervate individual muscle fibers. Of course, much of what skeletal muscle does isn't really conscious at all. When you walk, you're not consciously contracting and relaxing dozens of individual muscles in precise sequence. That's handled by motor programs in the spinal cord and lower brain regions. But you do have the ability to override these automatic patterns, to consciously control the contraction of most skeletal muscles if you choose to. Cardiac muscle, found exclusively in the heart, shares some structural features with skeletal muscle, but differs in critical ways. Like skeletal muscle, cardiac muscle is striated, reflecting a similar organization of contractile proteins. However, cardiac muscle cells, called cardiomyocytes, are shorter than skeletal muscle cells and typically contain only 1 or 2 nuclei per cell. More importantly, cardiac muscle cells are connected to each other through specialized junctions called intercalated discs, which contain both mechanical attachments called desmosomes and electrical connections called gap junctions. These gap junctions allow electrical signals to pass directly from one cardiac muscle cell to another, enabling the heart to contract as a coordinated unit. The control of cardiac muscle is involuntary. You cannot decide to make your heart beat faster or slower through conscious will, at least not directly. The heart generates its own rhythmic contractions through specialized pacemaker cells in the sinoatrial node, a property called autorhythmicity. These pacemaker cells spontaneously depolarize and fire action potentials at regular intervals, triggering waves of contraction that spread throughout the heart muscle. The autonomic nervous system can modulate the heart rate, speeding it up through sympathetic stimulation or slowing it down through parasympathetic stimulation, but it cannot override the heart's inherent rhythm entirely. Even if all nerve connections to the heart are severed, the heart continues to beat, though at a slower, intrinsic rate. Smooth muscle is the third type, found in the walls of hollow organs and tubes throughout the body. It lines the digestive tract from the esophagus to the rectum, forms the walls of blood vessels, controls the diameter of airways in the lungs, and can be found in the urinary bladder, the uterus, the eyes, and numerous other locations. Smooth muscle cells are smaller than skeletal or cardiac muscle cells, typically spindle-shaped, and contain a single nucleus positioned centrally within the cell. Unlike skeletal and cardiac muscle, smooth muscle lacks the regular geometric arrangement of contractile proteins that produces striations, giving it a smooth appearance under the microscope, hence the name. Smooth muscle is also involuntary, controlled by the autonomic nervous system, hormones, and local chemical factors. In many cases, smooth muscle exhibits automaticity, generating rhythmic contractions without external stimulation. The smooth muscle of the intestines, for example, produces slow, wave-like contractions called peristalsis that propel food through the digestive tract. These contractions are intrinsic to the muscle itself, though they can be modulated by neural and hormonal signals. The functional differences between these three muscle types reflect their different roles in the body. Skeletal muscle is built for power and speed, capable of generating large forces quickly, but also fatiguing relatively rapidly. Its voluntary control allows for precise, intentional movements. Cardiac muscle is built for endurance, contracting rhythmically and continuously for an entire lifetime without rest. Its autorhythmicity ensures that the heart keeps beating regardless of conscious control, a rather important feature for survival. Smooth muscle is built for sustained, low-level contractions, maintaining tension over long periods without fatigue. Its involuntary control allows it to regulate internal processes like digestion, blood pressure, and airway diameter without requiring conscious attention. The metabolic demands of muscle tissue are substantial. Even at rest, muscle tissue requires a continuous supply of energy to maintain ion gradients, synthesize proteins, and perform basic cellular housekeeping. During contraction, energy demands increase dramatically, sometimes by a factor of 100 or more. To meet these demands, muscle cells are packed with mitochondria, the cellular organelles responsible for aerobic energy production. Skeletal muscle fibers that specialize in endurance activities may have mitochondria occupying 25% or more of the XPO2 cell's volume, an extraordinary allocation of cellular resources to energy production. Muscle tissue also stores its own fuel supply in the form of glycogen, a branched polymer of glucose molecules. Glycogen granules are scattered throughout the cytoplasm of muscle cells, ready to be broken down rapidly when energy demands increase. A well-trained endurance athlete may store 400 to 500 grams of glycogen in their skeletal muscles, representing roughly 1,600 to 2,000 calories of stored energy. This is enough to fuel about 2 hours of moderate intensity exercise, after which the muscles must rely increasingly on fat as a fuel source. The blood supply to muscle tissue is extensive and highly regulated. Skeletal muscles receive blood through a network of arteries that branch into smaller arterioles and eventually into capillaries, the smallest blood vessels where gas and nutrient exchange occurs. The density of capillaries and muscle tissue varies depending on the fiber type, with endurance-oriented fibers having a much denser capillary network than power-oriented fibers. During rest, much of this capillary network is closed, with blood diverted to other tissues. During exercise, local metabolic signals cause the capillaries to open, dramatically increasing blood flow to the active muscles. This process, called exercise hyperemia, can increase blood flow to an active muscle by a factor of 20 or more. The nervous system's relationship with muscle tissue is intimate and essential. Without neural stimulation, skeletal muscle will not contract, period. The connection between nerve and muscle occurs at specialized synapses called neuromuscular junctions, sites where a motor neuron terminal comes into close contact with a muscle fiber's surface. When a nerve impulse reaches this junction, It triggers the release of a chemical messenger, acetylcholine, which binds to receptors on the muscle fiber and initiates contraction. This process, called excitation contraction coupling, is one of the most studied phenomena in physiology and will examine it in exhaustive detail in later chapters. Muscle tissue is not static. It adapts to the demands placed on it through processes of hypertrophy, the growth of existing muscle fibers, and atrophy, the shrinking of muscle fibers. When a muscle is used frequently and intensely, particularly in resistance exercise, the stress triggers cellular mechanisms that increase protein synthesis, causing the muscle fibers to grow thicker and stronger. Conversely, when a muscle is not used, when someone is bedridden or when a limb is immobilized in a cast, the muscle fibers shrink, losing both size and strength. This adaptability allows muscle tissue to optimize itself for the demands of the organism's lifestyle, but it also means that muscle mass and strength require continuous maintenance. The regenerative capacity of muscle tissue varies by type. Skeletal muscle has a limited ability to repair itself through the activation of satellite cells, a population of muscle stem cells that lie dormant along the muscle fibers. When muscle is damaged, these satellite cells activate, proliferate, and fuse with existing fibers to repair the damage. However, this regenerative capacity declines with age and is limited in extent. Severe muscle damage may result in the replacement of muscle tissue with scar tissue, which is non-contractile and represents a permanent loss of function. Cardiac muscle has even more limited regenerative capacity, with most cardiac muscle cells being unable to divide after birth. When cardiac muscle is damaged, as in a heart attack, the dead tissue is typically replaced with scar tissue, permanently reducing the heart's functional capacity. Smooth muscle falls somewhere in between, with some capacity for cell division and regeneration, though again, this capacity is limited. The evolution of muscle tissue represents one of the major innovations in the history of life on Earth. The earliest animals, simple multicellular organisms that appeared over 600 million years ago, likely had some form of contractile cells that allowed them to change shape or move through their environment. As animals became more complex, muscle tissue became more specialized, eventually differentiating into the distinct types we see today. The appearance of striated muscle, with its highly organized contractile apparatus, allowed for much more powerful and efficient movement, enabling the evolution of active predators, fast-moving prey, and eventually terrestrial locomotion. Without muscle tissue, animal life as we know it would be impossible. Understanding muscle tissue at a fundamental level provides the foundation for everything that follows. The basic properties of contractility, excitability, extensibility, and elasticity apply to all muscle types. The need for substantial energy supply, the dependence on nervous stimulation, the capacity for adaptation, all of these are features shared across muscle tissue types, even as the specific structures and control mechanisms differ. With this foundation established, we can now turn our attention to the microscopic architecture of muscle fibers, to the intricate structural organization that makes contraction possible. Because to understand how muscles generate force, we must first understand how they're built, from the level of the whole muscle down to the arrangement of individual protein molecules within the sarcomere. If you were to take a cross-section through a skeletal muscle and examine it under a microscope, you would immediately notice that muscle is not a uniform mass of tissue. Instead, it's highly organized, with distinct layers of connective tissue dividing the muscle into progressively smaller compartments. This organization is not merely aesthetic, it's functional, providing structural support, pathways for blood vessels and nerves, and a means of transmitting force from individual muscle fibers to the tendons that connect muscle to bone. The outermost layer of connective tissue surrounding an entire muscle is called the epimysium. This is a dense, irregular connective tissue composed primarily of collagen fibers, tough and relatively inelastic, providing a protective sheath around the muscle and helping to maintain its shape. The epimysium is continuous with the tendons at each end of the muscle, ensuring that the force generated by muscle contraction is effectively transmitted to the skeleton. Beneath the epimysium, the muscle is divided into bundles called fascicles. Each fascicle contains anywhere from a few dozen to several hundred individual muscle fibers, depending on the size and function of the muscle. The fascicles themselves are wrapped in another layer of connective tissue called the perimysium. Like the epimysium, the perimysium is composed primarily of collagen, though it's somewhat less dense. The perimysium contains the major blood vessels and nerves that supply the muscle, branching repeatedly as they penetrate into the fascicles to reach individual muscle fibers. Within each fascicle, individual muscle fibers are surrounded by a delicate layer of connective tissue called the endomysium. This is composed of a fine network of collagen and reticular fibers that wraps around each muscle fiber like a thin sleeve. The endomysium contains the capillaries that deliver oxygen and nutrients to the muscle fibers and remove metabolic waste products. It also provides a pathway for the motor neuron branches that stimulate muscle contraction. The muscle fibers themselves, the actual cells that contract, are remarkable structures. Unlike most cells in the body, which are small and contain a single nucleus, skeletal muscle fibers are enormous. They can be several centimeters long, sometimes extending the entire length of the muscle, and they can be up to 100 micrometers in diameter. More remarkably, each muscle fiber contains not one nucleus, but hundreds or even thousands of nuclei, distributed along the length of the fiber just beneath the cell membrane. This multinucleated state results from a unique developmental process. During embryonic development, individual myoblast cells, each with its own nucleus, fuse together to form a single, elongated muscle fiber. Once formed, the muscle fiber cannot divide. If you damage a muscle fiber badly enough to kill it, it cannot be replaced by the division of neighboring fibers. The only way new muscle fibers can be produced is through the fusion of satellite cells, a small population of muscle stem cells that persist in adult muscle tissue. These satellite cells remain quiescent under normal conditions but can be activated by muscle damage or intense exercise, after which they proliferate and fuse to repair or strengthen existing muscle fibers. The cell membrane of a muscle fiber is called the sarcolemma. This isn't just a simple lipid bilayer like the membrane of most cells. It's a complex structure that includes the plasma membrane itself, an overlying basement membrane, and associated proteins that connect the muscle fiber to the surrounding endomysium. The sarcolemma is critical for conducting electrical signals across the surface of the muscle fiber, signals that trigger contraction. It's also studded with various receptors and channels that allow the fiber to respond to hormones, metabolic signals, and mechanical stress. The cytoplasm of a muscle fiber is called the sarcoplasm because muscle physiologists, like most scientists, enjoy creating specialized terminology for familiar things. The sarcoplasm is densely packed with the machinery of contraction and energy production. Filling most of the available space are myofibrils, long cylindrical structures that run parallel to each other along the length of the fiber. These myofibrils are the contractile units of the muscle fiber, containing the proteins that generate force during contraction. Between and around the myofibrils, the sarcoplasm contains mitochondria, often in remarkable abundance. In muscle fibers specialized for endurance, the mitochondria may occupy 20 to 30% of the fiber's volume, packed in rows between myofibrils or clustered near the sarcolemma. These mitochondria are the powerhouses of the cell, using oxygen to convert nutrients into ATP, the energy currency that powers muscle contraction. The more mitochondria a muscle fiber has, the better it is at sustaining contraction over long periods without fatiguing. The sarcoplasm also contains glycogen granules, clusters of glucose molecules stored for rapid energy availability. When a muscle fiber begins to contract intensely, it can quickly break down glycogen into glucose and use it to fuel contraction, even before the blood supply can be increased to deliver more fuel. This stored glycogen is particularly important during the first few minutes of intense exercise, bridging the gap between resting metabolism and the full activation of aerobic energy production. Lipid droplets are another feature of the sarcoplasm, particularly in muscle fibers specialized for endurance. These droplets contain triglycerides, fats that can be broken down to fuel prolonged, moderate intensity contraction. While fat cannot be metabolized as quickly as carbohydrate, it provides much more energy per gram, making it an ideal fuel for sustained activity. The sarcoplasmic reticulum is perhaps the most distinctive feature of muscle fiber ultrastructure. This is an elaborate network of tubular membranes that surrounds each myofibril, forming a lacy sleeve around the contractile apparatus. The sarcoplasmic reticulum serves as the muscle fiber's calcium storage facility. In a resting muscle fiber, the sarcoplasmic reticulum actively pumps calcium ions out of the sarcoplasm and stores them at high concentrations within its interior. When the muscle fiber is stimulated to contract, the sarcoplasmic reticulum releases this stored calcium into the sarcoplasm, where it triggers the contractile process. When contraction is complete, the sarcoplasmic reticulum pumps the calcium back into storage, allowing the muscle to relax. The sarcoplasmic reticulum is organized into two distinct regions. The longitudinal tubules run parallel to the myofibrils, forming the bulk of the network. At regular intervals, the longitudinal tubules expand into enlarged chambers called terminal cisternae. These terminal cisternae are positioned adjacent to another membrane system, the transverse tubules, or tubules. Tubules are invaginations of the sarcolemma, narrow tubes that plunge from the surface of the muscle fiber deep into its interior. These tubules ensure that electrical signals traveling across the sarcolemma can rapidly penetrate into the depths of the muscle fiber, reaching myofibrils that would otherwise be too far from the surface to be activated quickly. The tubules run perpendicular to the myofibrils, typically positioned at specific locations that we'll identify shortly. Where a tubule passes between two terminal cisternae of the sarcoplasmic reticulum, the three structures form a complex called a triad. The triad is the functional unit for excitation-contraction coupling, the process by which an electrical signal is converted into a mechanical contraction. When an action potential travels down a tubule, it triggers the release of calcium from the adjacent terminal cisternae, initiating contraction in the nearby myofibril. Because tubules penetrate throughout the muscle fiber, connecting to triads positioned at regular intervals along every myofibril, a single action potential on the sarcolemma can trigger nearly simultaneous contraction throughout the entire fiber. Now we come to the myofibrils themselves, the actual contractile apparatus of the muscle fiber. Each myofibril is about 1 micrometer in diameter and extends the full length of the muscle fiber. A single muscle fiber may contain thousands of myofibrils, packed so tightly that they occupy the majority of the fiber's volume. Under an electron microscope, myofibrils display a remarkable striated appearance, alternating light and dark bands that repeat along their length. This striation is the hallmark of skeletal and cardiac muscle, and it reflects the precise geometric organization of the contractile proteins within the myofibril. The repeating unit of this organization is called the sarcomere. Each sarcomere extends from one Z-disc to the next Z-disc, a distance of about 2 to 2.5 micrometers in a resting muscle. The sarcomere is the fundamental contractile unit of muscle. When a muscle contracts, each sarcomere shortens, and because thousands of sarcomeres are arranged in series along the length of each myofibril, their combined shortening produces the overall contraction of the muscle fiber. The Z-disc, named from the German word zwischen, meaning between, is a dense protein structure that forms the boundary between adjacent sarcomeres. In electron micrographs, the Z-disc appears as a dark, zigzagging line running perpendicular to the long axis of the myofibril. The Z-disc serves as an anchor point for thin filaments, one of the 2 primary types of protein filaments that generate muscle contraction. Extending from each side of the Z-disc are the thin filaments, composed primarily of the protein actin. These filaments are about 7 nanometers in diameter and roughly 1 micrometer in length. They extend from the Z-disc toward the center of the sarcomere, where they interdigitate with the thick filaments. The thick filaments are composed primarily of the protein myosin. These filaments are about 15 nanometers in diameter and 1.6 micrometers long. They are positioned in the center of the sarcomere, running parallel to the thin filaments. The thick filaments are connected to each other by proteins at the M line, a structure in the very center of the sarcomere that helps maintain the geometric arrangement of the thick filaments. The arrangement of these filaments creates the striated appearance of the myofibril. The dark bands, called A bands, A for anisotropic, referring to their optical properties under polarized light, correspond to the length of the thick filaments. The A band includes both the thick filaments and the overlapping portions of the thin filaments, giving it a dark appearance due to the high protein density. The light bands, called I bands, I for isotropic, contain only thin filaments and the Z-disc, and they appear lighter because the protein density is lower. Within the A band, there is a lighter region called the H zone. This corresponds to the central portion of the thick filaments, where there is no overlap with thin filaments. The H zone narrows during contraction as the thin filaments slide deeper into the A band, and it can disappear entirely during maximal contraction. In the center of the H zone is the M line, where proteins link adjacent thick filaments together, maintaining their parallel alignment. The geometric precision of this arrangement is remarkable. In a single sarcomere, there are approximately 1,500 thick filaments, each surrounded by 6 thin filaments in a hexagonal array. This geometric arrangement ensures that each thick filament is optimally positioned to interact with the surrounding thin filaments during contraction. Multiply this by the thousands of sarcomeres in each myofibril and the millions of myofibrils in a muscle, and you begin to appreciate the staggering complexity of the contractile apparatus. But the sarcomere contains more than just actin and myosin. There are numerous accessory proteins that maintain the structure of the sarcomere, regulate contraction, and connect the contractile apparatus to other cellular structures. One of the most important of these is titin, often called the third filament, though it's quite different from actin and myosin. Titin is an enormous protein, the largest known in the human body, with a molecular weight exceeding 3 million daltons. A single titin molecule extends from the Z disc to the M line, spanning half the length of the sarcomere. Titin serves multiple functions. In the I band region, it acts as a molecular spring, providing passive tension that helps the sarcomere return to its resting length after being stretched. In the A band region, it helps anchor the thick filaments to the Z disc and maintains the centering of the thick filaments within the sarcomere. Titin also contains binding sites for numerous other proteins and plays a role in sensing mechanical stress and triggering adaptive responses in the muscle fiber. Another important accessory protein is nebulin, which extends along the length of the thin filaments from the Z disc. Nebulin acts as a molecular ruler, determining the length of the thin filaments during sarcomere assembly. It also appears to play a role in stabilizing the thin filaments and regulating their interaction with myosin. The Z disc itself contains numerous proteins that anchor the thin filaments and connect adjacent sarcomeres to each other and to the sarcolemma. Alpha-actinin is the primary structural protein of the Z disc, forming a lattice-like structure that provides anchoring sites for the thin filaments. Other Z-disc proteins link the myofibrils to each other, ensuring that they contract in synchrony, and to the costomeres, protein complexes that connect the myofibrils to the sarcolemma and ultimately to the surrounding endomysium. This hierarchical organization, from the whole muscle down to the individual sarcomere, ensures that the force generated by molecular interactions within the sarcomere is efficiently transmitted to the skeleton. When myosin pulls on actin within a sarcomere, that force is transmitted through the Z disc to adjacent sarcomeres, through the costomeres to the sarcolemma, through the endomysium to the perimysium, and through the perimysium to the epimysium, and finally to the tendons. At each level of organization, the structural proteins are arranged to minimize energy loss and maximize force transmission. The number of sarcomeres in series along a myofibril determines how much the muscle fiber can shorten. Each sarcomere can shorten by about 50% during maximal contraction, from roughly 2.5 micrometers to 1.2 micrometers. If a myofibril contains 10,000 sarcomeres, which is typical for a muscle fiber that's a few centimeters long, then the myofibril can shorten by 10,000 times 1.3 micrometers, or 13 millimeters. This explains why longer muscles can produce greater ranges of motion than shorter muscles, even if they generate the same force. The number of myofibrils in parallel within a muscle fiber, combined with the number of muscle fibers in parallel within a muscle, determines the maximum force the muscle can generate. Each sarcomere generates a small amount of force, but because millions of sarcomeres are acting in parallel, the forces sum to produce the substantial pulling forces that muscles can generate. A muscle with larger cross-sectional area contains more myofibrils in parallel and can therefore generate more force. This architecture also explains why muscle has direction. Muscle can only pull, it cannot push. The sliding of thin filaments past thick filaments can only draw the Z discs closer together, shortening the sarcomere. To achieve coordinated movement, muscles must be arranged in opposing pairs or groups, with one muscle or group pulling in one direction and another pulling in the opposite direction. We'll explore these arrangements in detail in later chapters. The microscopic architecture of smooth muscle differs considerably from that of skeletal muscle, lacking the precise geometric organization that produces striations. Smooth muscle cells are much smaller, typically only 50 to 400 micrometers long and 5 to 10 micrometers in diameter. They contain a single, centrally positioned nucleus. The contractile proteins in smooth muscle are organized differently, with actin and myosin filaments arranged in a less regular pattern and anchored to structures called dense bodies scattered throughout the cell rather than to Z discs. This arrangement still allows for contraction through the sliding of actin past myosin, but the mechanism is regulated differently, as we'll see in a later chapter. Cardiac muscle falls somewhere between skeletal and smooth muscle in its organization. Like skeletal muscle, cardiac muscle is striated, with sarcomeres organized into myofibrils. However, cardiac muscle cells are much shorter than skeletal muscle fibers, typically only 100 micrometers long, and they're branched, allowing each cell to connect to several neighbors. The connections between cardiac muscle cells, the intercalated discs, contain both mechanical junctions that transmit force and electrical junctions that allow action potentials to spread from cell to cell. This organization allows the heart to contract as a coordinated unit, with waves of contraction spreading across the heart muscle. Understanding this microscopic architecture is essential to understanding how muscles contract. The precision of the sarcomere organization, the arrangement of myofibrils within fibers, the organization of fibers into fascicles, all of this structural hierarchy exists to serve the fundamental function of converting chemical energy into mechanical force. Now that we've established the structural framework, we can turn our attention to the molecular players, the individual proteins whose interactions generate the forces that allow muscles to move the body. Muscle contraction, at its most fundamental level, is a story about proteins. Not the proteins you eat for breakfast, though those are certainly related, but rather the specialized protein molecules that make up the contractile machinery within muscle fibers. These proteins have been refined by hundreds of millions of years of evolution into molecular machines of remarkable efficiency and precision. Understanding how they work requires us to zoom in even further than we did in the previous chapter, down to the level of individual molecules and the forces that govern their interactions. Let's begin with actin, the primary component of the thin filaments. Actin is a globular protein, roughly spherical in shape, with a molecular weight of about 42,000 daltons. In its monomeric form, it's called G-actin, G for globular. Each G-actin molecule has a binding site for ATP or ADP, and in living cells, it's usually found with ADP bound. G-actin also has a binding site for myosin, the protein that will actually pull on it during contraction, though in a resting muscle, this binding site is blocked by other proteins. G-actin molecules don't remain as individual units. They polymerize, linking together in long chains to form filamentous actin, or F-actin. Each F-actin filament looks like a string of beads twisted into a double helix, with the helix completing one full turn every 13 or 14 actin monomers. The thin filament of the sarcomere is essentially two of these F-actin chains wound around each other, creating a helical structure about 7 nanometers in diameter and roughly 1 micrometer in length. The two F-actin strands form a groove between them, a helical groove that spirals around the thin filament. Sitting in this groove are molecules of another protein called tropomyosin. Tropomyosin is a rod-shaped protein about 40 nanometers long, composed of two alpha helical chains wound around each other. Each tropomyosin molecule spans about 7 actin monomers, and successive tropomyosin molecules are arranged end to end along the thin filament, effectively covering the entire length of the F-actin helix. The function of tropomyosin is essentially regulatory. In a resting muscle, tropomyosin sits in a position that blocks the myosin binding sites on actin. This prevents myosin from attaching to actin and generating force. For contraction to occur, tropomyosin must be moved out of the way, exposing the myosin binding sites. This is where the third major component of the thin filament comes into play: troponin. Troponin is actually a complex of 3 different proteins, each with a specific role. Troponin T binds to tropomyosin, anchoring the troponin complex to the thin filament. Troponin the first binds to actin, helping to maintain tropomyosin's blocking position in a resting muscle. And troponin C, the most critical of the 3, has binding sites for calcium ions. Here's how the regulatory mechanism works. In a resting muscle fiber, the concentration of calcium in the sarcoplasm is extremely low, about 0.1 micromolar, roughly 10,000 times lower than the concentration in the sarcoplasmic reticulum. At this low calcium concentration, troponin C has no calcium bound to it. Without calcium, the troponin complex holds tropomyosin in a position that blocks the myosin binding sites on actin. The muscle remains relaxed, not because myosin can't work, but because it can't access actin. When the muscle fiber is stimulated and calcium floods into the sarcoplasm, calcium ions bind to troponin C. Each troponin C molecule has 4 calcium binding sites, though typically only 2 of them are involved in regulating contraction. When calcium binds, it causes a conformational change in the troponin complex. Troponin the first releases its inhibitory hold on actin, and the entire troponin-tropomyosin complex shifts position, rolling slightly deeper into the groove between the 2 F-actin chains. This movement exposes the myosin binding sites on actin, allowing contraction to proceed. One troponin complex sits on the thin filament about every 40 nanometers, positioned over every 7th actin monomer. Because each troponin complex controls the position of one tropomyosin molecule, and each tropomyosin molecule spans 7 actin monomers, one troponin complex effectively regulates the availability of 7 myosin binding sites. This provides an efficient amplification mechanism. Binding of a few calcium ions to troponin C can expose multiple binding sites for myosin. Now let's turn to myosin, the molecular motor that actually generates force. Myosin is a much larger and more complex protein than actin. The myosin molecule found in skeletal muscle, called myosin 2, has a molecular weight of about 500,000 daltons. It consists of 6 polypeptide chains, 2 heavy chains and 4 light chains. Each heavy chain has a globular head region and a long rod-like tail. The 2 heavy chains wrap around each other in the tail region, forming an alpha helical coil, much like 2 pieces of rope twisted together. This tail is about 150 nanometers long and quite stiff, providing the structural backbone of the thick filament. At one end of the tail, the 2 heavy chains diverge, each forming a globular head region. So each complete myosin molecule has 2 heads, each capable of binding to actin and generating force. The globular head region of myosin is where the action happens. This head contains a binding site for actin and a binding site for ATP. It also contains the enzymatic machinery to hydrolyze ATP, breaking it down into ADP and inorganic phosphate, releasing energy in the process. This makes myosin not just a structural protein, but an enzyme, specifically an ATPase. The head also contains the molecular machinery to convert the chemical energy from ATP hydrolysis into mechanical movement, a process called mechanochemical transduction. Associated with each myosin head are two light chains, small proteins that wrap around the neck region where the head connects to the tail. These light chains play regulatory and structural roles, blocked by other proteins. G-actin molecules don't remain as individual units. They polymerize, linking together in long chains to form filamentous actin, or F-actin. Each F-actin filament looks like a string of beads twisted into a double helix, with the helix completing one full turn every 13 or 14 actin monomers. The thin filament of the sarcomere is essentially two of these F-actin chains wound around each other, creating a helical structure about 7 nanometers in diameter and roughly 1 micrometer in length. The two F-actin strands form a groove between them, a helical groove that spirals around the thin filament. Sitting in this groove are molecules of another protein called tropomyosin. Tropomyosin is a rod-shaped protein about 40 nanometers long, composed of two alpha helical chains wound around each other. Each tropomyosin molecule spans about 7 actin monomers, and successive tropomyosin molecules are arranged end to end along the thin filament, effectively covering the entire length of the F-actin helix. The function of tropomyosin is essentially regulatory. In a resting muscle, tropomyosin sits in a position that blocks the myosin binding sites on actin. This prevents myosin from attaching to actin and generating force. For contraction to occur, tropomyosin must be moved out of the way, exposing the myosin binding sites. This is where the third major component of the thin filament comes into play, troponin. Troponin is actually a complex of 3 different proteins, each with a specific role. Troponin T binds to tropomyosin, anchoring the troponin complex to the thin filament. Troponin I binds to actin, helping to maintain tropomyosin's blocking position in a resting muscle. And troponin C, the most critical of the 3, has binding sites for calcium ions. Here's how the regulatory mechanism works. In a resting muscle fiber, the concentration of calcium in the sarcoplasm is extremely low, about 0.1 micromolar, roughly 10,000 times lower than the concentration in the sarcoplasmic reticulum. At this low calcium concentration, troponin C has no calcium bound to it. Without calcium, the troponin complex holds tropomyosin in a position that blocks the myosin binding sites on actin. The muscle remains relaxed, not because myosin can't work, but because it can't access actin. When the muscle fiber is stimulated and calcium floods into the sarcoplasm, calcium ions bind to troponin C. Each troponin C molecule has 4 calcium binding sites, though typically only 2 of them are involved in regulating contraction. When calcium binds, it causes a conformational change in the troponin complex. Troponin I releases its inhibitory hold on actin, and the entire troponin-tropomyosin complex shifts position, rolling slightly deeper into the groove between the 2 F-actin chains. This movement exposes the myosin binding sites on actin, allowing contraction to proceed. One troponin complex sits on the thin filament about every 40 nanometers, positioned over every seventh actin monomer. Because each troponin complex controls the position of one tropomyosin molecule, and each tropomyosin molecule spans 7 actin monomers, one troponin complex effectively regulates the availability of 7 myosin binding sites. This provides an efficient amplification mechanism. Binding of a few calcium ions to troponin C can expose multiple binding sites for myosin. Now let's turn to myosin, the molecular motor that actually generates force. Myosin is a much larger and more complex protein than actin. The myosin molecule found in skeletal muscle, called myosin 2, has a molecular weight of about 500,000 daltons. It consists of 6 polypeptide chains, 2 heavy chains, and 4 light chains. Each heavy chain has a globular head region and a long, rod-like tail. The 2 heavy chains wrap around each other in the tail region, forming an alpha helical coil, much like 2 pieces of rope twisted together. This tail is about 150 nanometers long and quite stiff, providing the structural backbone of the thick filament. At one end of the tail, the 2 heavy chains diverge, each forming a globular head region. So each complete myosin molecule has 2 heads, each capable of binding to actin and generating force. The globular head region of myosin is where the action happens. This head contains a binding site for actin and a binding site for ATP. It also contains the enzymatic machinery to hydrolyze ATP, breaking it down into ADP, an inorganic phosphate, releasing energy in the process. This makes myosin not just a structural protein, but an enzyme, specifically an ATPase. The head also contains the molecular machinery to convert the chemical energy from ATP hydrolysis into mechanical movement, a process called mechanochemical transduction. Associated with each myosin head are 2 light chains, small proteins that wrap around the neck region where the head connects to the tail. These light chains play regulatory and structural roles. These glycoproteins, in turn, connect to laminin in the basement membrane outside the cell. This creates a continuous mechanical link from the contractile apparatus through the cell membrane to the extracellular matrix. The importance of dystrophin becomes tragically apparent in Duchenne muscular dystrophy, a genetic disorder caused by mutations in the dystrophin gene. Without functional dystrophin, the connection between the myofibrils and the sarcolemma is weakened. During contraction, the force generated by the myofibrils can tear the sarcolemma, leading to progressive damage and degeneration of the muscle fibers. Children with Duchenne muscular dystrophy typically begin to show symptoms around age 3 to 5, with progressive muscle weakness that eventually affects the respiratory and cardiac muscles. Most patients do not survive beyond their 20s or 30s, a stark reminder of the critical importance of the structural proteins that we usually take for granted. In smooth muscle, the organization of the contractile proteins is quite different. Smooth muscle lacks the precise geometric organization of sarcomeres, so it doesn't have Z-discs or M-lines. Instead, actin filaments are anchored to structures called dense bodies, which are scattered throughout the cell. Some dense bodies are attached to the cell membrane, while others float freely in the cytoplasm, connected to each other and to the membrane-associated dense bodies by intermediate filaments. The actin filaments in smooth muscle are arranged in a loose lattice that crisscrosses the cell. Thick filaments, composed of myosin, are interspersed among the actin filaments, though they're present in lower numbers than in skeletal muscle. When smooth muscle contracts, the myosin pulls on the actin filaments, which in turn pull on the dense bodies. This causes the cell to contract in all directions, rather like pulling the drawstrings on a pouch, causing the entire cell to shorten and generate force. The regulation of smooth muscle contraction also differs from skeletal muscle. While skeletal muscle uses the troponin-tropomyosin system to regulate the availability of myosin-binding sites on actin, smooth muscle uses a different mechanism that regulates myosin's ability to interact with actin. In smooth muscle, myosin can only bind to actin and generate force when one of its light chains, called the regulatory light chain, is phosphorylated. This phosphorylation is carried out by an enzyme called myosin light chain kinase, which is activated by calcium binding to a protein called calmodulin. So in smooth muscle, calcium still plays the critical regulatory role, but it acts through a different mechanism. When calcium levels rise, calcium binds to calmodulin, activating myosin light chain kinase, which phosphorylates myosin, allowing it to interact with actin. When calcium levels fall, a phosphatase enzyme removes the phosphate groups from myosin, inactivating it. This phosphorylation-dephosphorylation cycle is slower than the troponin-tropomyosin system of skeletal muscle, which is part of why smooth muscle contracts more slowly but can maintain contraction for much longer periods without fatigue. Cardiac muscle uses a contractile apparatus very similar to that of skeletal muscle, with sarcomeres organized in the same way and regulated by the same troponin-tropomyosin system. However, cardiac muscle has some unique features. The thin filaments in cardiac muscle are slightly shorter than in skeletal muscle, and the troponin isoforms are different, with different calcium sensitivities. These differences affect the force-generating capacity and the calcium sensitivity of cardiac muscle, optimizing it for the continuous, rhythmic contractions of the heart. The molecular machinery of muscle contraction is, in many ways, one of the triumphs of evolution. These proteins have been refined over hundreds of millions of years, optimized for efficiency, speed, and reliability. The fact that myosin can convert the chemical energy of ATP into mechanical work with an efficiency approaching 50% is remarkable. The fact that this conversion happens on timescales of milliseconds, with forces generated and released hundreds of times per second during rapid movements, is even more remarkable. And the fact that this machinery can function reliably for decades, with cardiac muscle contracting billions of times over a human lifetime without replacement, is perhaps most remarkable of all. But proteins, no matter how sophisticated, cannot function without energy. The ATP that powers myosin's molecular motor must come from somewhere, and muscle tissue has extraordinary energy demands, particularly during intense activity. So now we must turn our attention to the metabolic machinery that fuels muscle contraction, to the pathways that convert nutrients into ATP, and the systems that ensure ATP is available whenever and wherever it's needed within the muscle fiber. ATP, adenosine triphosphate, is often called the energy currency of the cell, and nowhere is this metaphor more apt than in muscle tissue. Every molecular event in muscle contraction, from the binding and release of myosin to actin to the pumping of calcium back into the sarcoplasmic reticulum to the maintenance of ion gradients across the sarcolemma, requires ATP. The demand for ATP in active muscle is staggering, and understanding how muscle meets this demand requires a journey through cellular metabolism from the immediate energy systems that power the first few seconds of contraction to the aerobic pathways that sustain prolonged activity. Let's begin with the ATP molecule itself. ATP consists of adenosine, a molecule composed of the nitrogenous base adenine attached to the sugar ribose, with three phosphate groups attached in a chain. The bonds between these phosphate groups, particularly the bond between the second and third phosphate groups, are called high-energy bonds. This terminology is slightly misleading. The bonds themselves don't contain energy in the way that gasoline contains energy. Rather, when these bonds are broken through hydrolysis, the reaction releases energy because the products, ADP and inorganic phosphate, are at a lower energy state than ATP. When myosin hydrolyzes ATP, breaking it down into ADP and inorganic phosphate, about 7.3 kilocalories per mole of energy is released under standard conditions, though in the cellular environment, the actual free energy change is closer to 12 kilocalories per mole. This energy is used to change the conformation of the myosin head, cocking it into a high-energy position ready to perform the power stroke. The power stroke itself occurs when the myosin head releases the inorganic phosphate and then the ADP, pulling on the actin filament as it returns to its low-energy conformation. A resting muscle contains only enough ATP to fuel about 2 to 3 seconds of maximal contraction. If you were to begin lifting a weight, your muscles would deplete their stored ATP almost immediately. Yet obviously, you can continue contracting your muscles for much longer than a few seconds. This is because muscle fibers have multiple systems for regenerating ATP as quickly as it's consumed. These systems operate on different time scales, with the fastest systems providing energy for the first few seconds of activity, and slower but more sustainable systems taking over for prolonged contraction. The most immediate system is the phosphocreatine system, also called the creatine phosphate system. Muscle fibers contain about 4 to 6 times as much phosphocreatine as ATP. Phosphocreatine is a high-energy molecule with a phosphate group attached to creatine via a high-energy bond, similar to those in ATP. When ATP is consumed during contraction, an enzyme called creatine kinase rapidly transfers the phosphate group from phosphocreatine to ADP, regenerating ATP. This reaction is extremely fast, fast enough to keep ATP concentrations nearly constant during the first few seconds of intense muscle activity. The phosphocreatine system doesn't require oxygen, doesn't produce lactate, and doesn't rely on complex metabolic pathways. It's simply a quick chemical shuffle, moving phosphate groups from one molecule to another. This makes it ideal for powering short bursts of maximal effort, the kind of effort involved in throwing a punch, jumping, or lifting a heavy weight. However, the phosphocreatine stores are limited, enough to power about 10 to 15 seconds of maximal effort. Once phosphocreatine is depleted, muscle must rely on other systems to regenerate ATP. The next system to engage is anaerobic glycolysis. Glycolysis is the metabolic pathway that breaks down glucose into pyruvate, yielding a small amount of ATP in the process. The pathway consists of 10 enzyme-catalyzed reactions taking place in the cytoplasm of the cell. Each glucose molecule is broken down into 2 3-carbon pyruvate molecules, yielding a net production of 2 ATP molecules. This might not seem like much, but glycolysis can proceed extremely rapidly, faster than aerobic metabolism, making it valuable for meeting the high ATP demands of intense muscle activity. The glucose for glycolysis can come from 2 sources, It can be delivered to the muscle via the bloodstream, or it can come from glycogen stored within the muscle fiber itself. Glycogen is a branched polymer of glucose molecules, and muscle fibers store it in granules scattered throughout the sarcoplasm. When the muscle needs glucose quickly, enzymes break down glycogen into individual glucose molecules, a process called glycogenolysis. A well-trained athlete's muscles can store about 400 to 500 grams of glycogen, representing roughly 1,600 to 2,000 calories of stored energy. The accumulation of lactate in muscle has long been blamed for muscle fatigue and the burning sensation during intense exercise. However, more recent research suggests this view is oversimplified. Lactate itself isn't particularly harmful. In fact, it can be used as a fuel source by other muscles, by the heart, and by the liver, which can convert it back to glucose. The burning sensation during intense exercise is more likely related to the accumulation of hydrogen ions, which lower the pH of the muscle and can interfere with various cellular processes, including the calcium sensitivity of troponin and the function of glycolytic enzymes. Anaerobic glycolysis can sustain high-intensity muscle activity for about 1 to 3 minutes. During this time, the muscle is essentially running up an oxygen debt, consuming ATP faster than it can be regenerated through aerobic metabolism. Eventually, either the muscle must reduce its intensity, allowing aerobic metabolism to catch up, or the accumulation of metabolic byproducts and the depletion of glycogen will cause fatigue and force a reduction in activity. For sustained muscle activity, aerobic metabolism is essential. This is the domain of the mitochondria, the cellular powerhouses that can extract far more energy from nutrients than glycolysis alone. Aerobic metabolism can completely oxidize glucose, fatty acids, and even amino acids to carbon dioxide and water, capturing the released energy as ATP. The process begins with pyruvate, produced by glycolysis, being transported into the mitochondria. There, an enzyme complex called pyruvate dehydrogenase converts pyruvate to acetyl-CoA, removing a carbon dioxide molecule in the process. The acetyl-CoA enters the citric acid cycle, also known as the Krebs cycle or TCA cycle, a series of 8 enzyme-catalyzed reactions that completely oxidize the acetyl group to carbon dioxide. The citric acid cycle itself produces only a small amount of ATP directly, 1 molecule per turn of the cycle. However, it produces large amounts of NADH and FADH, 2 electron carriers that donate electrons to the electron transport chain on the inner mitochondrial membrane. As electrons flow through the transport chain, energy is released and used to pump protons across the inner mitochondrial membrane, creating an electrochemical gradient. When these protons flow back across the membrane through ATP synthase, the energy drives the synthesis of ATP from ADP and inorganic phosphate. This process, called oxidative phosphorylation, is remarkably efficient. The complete aerobic oxidation of 1 glucose molecule yields about 32 ATP molecules, compared to the 2 ATP from glycolysis alone. This 16-fold increase in efficiency is why aerobic metabolism is essential for sustained activity. However, aerobic metabolism requires oxygen delivered to the muscle via the bloodstream, and it requires time for the metabolic pathways to ramp up to full capacity. During the first few minutes of exercise, even if oxygen is available, aerobic metabolism cannot meet the full ATP demand of the muscle. It takes time for blood flow to increase, for mitochondrial enzymes to activate, and for the concentration of metabolic intermediates to build up. This is why even aerobic activities begin with an oxygen deficit, with the muscle relying on phosphocreatine and anaerobic glycolysis to bridge the gap until aerobic metabolism can fully engage. As exercise continues, aerobic metabolism gradually takes over as the primary ATP source. For moderate intensity exercise, aerobic metabolism can meet the muscle's ATP needs indefinitely, limited only by the availability of fuel and the accumulation of fatigue factors unrelated to ATP production. After exercise ceases, oxygen consumption remains elevated for a period, sometimes for hours, as the body pays back the oxygen debt, replenishing phosphocreatine stores, clearing lactate, and repairing cellular damage. For very long duration exercise, like running a marathon, the muscle must shift its fuel source from primarily carbohydrates to increasingly relying on fats. This is because glycogen stores are limited, but fat stores are essentially unlimited, even in lean individuals. A person with 15% body fat has tens of thousands of calories stored as triglycerides in adipose tissue, far more than the 1,600 to 2,000 calories stored as glycogen. However, fat metabolism is slower than carbohydrate metabolism. Breaking down triglycerides into fatty acids and glycerol, transporting the fatty acids into mitochondria, and oxidizing them through beta oxidation, and the citric acid cycle takes time and requires more oxygen per ATP produced than carbohydrate metabolism. You can sprint at maximal intensity for only 10 to 15 seconds, run at high intensity for perhaps a few minutes, jog at moderate intensity for an hour or more, and walk at low intensity for many hours, with the limitation at each intensity being the rate at which ATP can be supplied. Type 2A fibers are intermediate, with moderate mitochondrial content and the capacity for both aerobic and anaerobic metabolism. They can sustain activity longer than type 2X fibers, but generate force more quickly than type 1 fibers, making them versatile for a range of activities. Beyond ATP, muscle contraction requires the maintenance of ion gradients, particularly for calcium and sodium. The sarcoplasmic reticulum actively pumps calcium out of the sarcoplasm using ATP-dependent calcium pumps, about 2 calcium ions transported per ATP hydrolyzed. In a resting muscle, this represents a small ATP cost, but during repeated contractions, when calcium must be pumped back and forth with each contraction relaxation cycle, the ATP cost becomes substantial. Similarly, maintaining the sodium and potassium gradients across the sarcolemma requires ATP-dependent sodium-potassium pumps. During intense activity, the ATP cost of maintaining ion gradients can be as much as 30% of the total ATP consumption, with the remaining 70% going to fuel cross-bridge cycling. This is one reason why muscle efficiency, defined as mechanical work output divided by metabolic energy input, is typically only 20 to 25%. The majority of metabolic energy is converted to heat rather than mechanical work, which is why intense exercise raises body temperature and why muscles feel warm to the touch during activity. The heat production is not entirely wasteful. It helps maintain muscle temperature, and since biochemical reactions proceed faster at higher temperatures, this actually enhances muscle performance to a point. This is why warming up before intense activity improves performance. The increased muscle temperature speeds up enzyme activity, reduces muscle stiffness, and enhances the rate of cross-bridge cycling. However, if muscle temperature rises too high, above about 40 degrees Celsius, protein denaturation and enzyme dysfunction begin to occur, impairing performance. After exercise, muscle must recover, replenishing energy stores and repairing cellular damage. Phosphocreatine stores are replenished within a few minutes using ATP generated through aerobic metabolism. Glycogen replenishment takes longer, typically 24 to 48 hours, depending on the extent of depletion and carbohydrate intake. If glycogen stores are severely depleted and carbohydrate intake is insufficient, muscles may remain depleted for days, leading to persistent fatigue. The metabolic machinery of muscle is adaptive, responding to training by increasing the quantity and efficiency of the systems that are stressed. Endurance training increases mitochondrial biogenesis, the production of new mitochondria, and upregulates the expression of aerobic enzymes. Capillary density increases, enhancing oxygen delivery. These adaptations shift the muscle toward greater reliance on aerobic metabolism, improving endurance capacity. Conversely, detraining or inactivity leads to mitochondrial loss and reduced aerobic capacity, changes that can occur within weeks. Sprint training, on the other hand, enhances anaerobic capacity. Glycolytic enzyme levels increase, glycogen storage capacity expands, and buffering capacity improves, allowing the muscle to better tolerate the accumulation of hydrogen ions. While these adaptations don't increase the absolute duration that anaerobic metabolism can sustain activity, they do allow higher power outputs for the time it lasts. The metabolic flexibility of muscle, its ability to switch between fuel sources and metabolic pathways depending on intensity, duration, and fuel availability, is one of its most remarkable features. This flexibility allows humans to perform an extraordinary range of physical activities, from the explosive power of a sprint to the sustained endurance of a multi-hour run, from the repetitive contractions of chewing to the steady tension of maintaining posture. But metabolism is only part of the story. The ATP that muscle metabolism produces must be used effectively, converted from chemical energy into mechanical force through the sliding filament mechanism. And so we now turn to the cross-bridge cycle, the molecular dance between myosin and actin that actually generates the forces that move the body. The sliding filament theory, first proposed in 1954 by Andrew Huxley and Rolf Niedergerke, and independently by Hugh Huxley and Gene Hansen, stands as one of the landmark discoveries in physiology. Before this theory, the mechanism of muscle contraction was a mystery. Various proposals had been put forward, including the idea that muscle proteins folded like a spring or that they underwent some sort of chemical contraction. The breakthrough came from careful microscopic observations of muscle fibers during contraction, which revealed that the lengths of the individual filaments didn't change. Instead, the filaments slid past each other, with thin filaments being pulled toward the center of the sarcomere. This sliding is driven by the cyclical interaction between myosin heads and actin, a process called cross-bridge cycling. The term cross-bridge refers to the connection formed when a myosin head binds to actin, literally bridging between the thick and thin filaments. During each cycle, a myosin head binds to actin, pulls, releases, and then repositions itself to bind again. When hundreds of myosin heads are cycling asynchronously along a thick filament, the result is a sustained pull on the thin filaments, drawing them toward the center of the sarcomere. Let's walk through the cross-bridge cycle in detail, step by step. We'll begin with a myosin head that has just completed a power stroke and is bound to actin. At this point, the myosin head is in a low-energy conformation, bent at an angle like an oar at the end of a rowing stroke. The myosin-actin binding is quite strong, strong enough that without further input, the myosin would remain bound indefinitely. This is actually the state muscle enters in rigor mortis, when ATP is no longer available and myosin heads become locked onto actin, unable to detach. The first step in the cycle is the binding of ATP to the myosin head. The myosin head has a binding site for ATP on its surface, distinct from the actin binding site. When an ATP molecule binds to this site, it causes a conformational change in the myosin head that dramatically reduces the affinity of myosin for actin. In practical terms, the ATP binding causes the myosin to let go of actin. This detachment occurs within milliseconds of ATP binding. Now the myosin head is detached from actin, and the second step of the cycle can occur, ATP hydrolysis. The myosin head contains ATPase activity, meaning it can break down ATP into ADP and inorganic phosphate. This hydrolysis reaction releases energy, and that energy is used to change the conformation of the myosin head, cocking it into a high energy position. Think of this like cocking the hammer on a gun or drawing back the string on a bow. The myosin head pivots at its neck region, rotating to a more perpendicular angle relative to the thick filament. The ADP and inorganic phosphate remain bound to the myosin head at this stage, though they're bound loosely. In this cocked high-energy position, the myosin head is ready to bind to actin again. This is the third step, cross-bridge formation. The myosin head, still holding ADP and phosphate, reaches out and binds to an actin molecule on the thin filament. However, it can only do this if the myosin binding site on actin is exposed, which, as we discussed in the previous chapter, only happens when calcium is present and the troponin-tropomyosin complex has shifted to uncover the binding sites. The binding of myosin to actin triggers the fourth step, the power stroke. When the myosin head attaches to actin, it causes a conformational change that releases the inorganic phosphate. This release of phosphate triggers another conformational change in the myosin head, causing it to pivot back toward its original low-energy position. As it pivots, it pulls on the actin filament, dragging it toward the center of the sarcomere. This is the power stroke, the actual force-generating event of muscle contraction. The power stroke moves the actin filament about 10 nanometers, a tiny distance at the molecular scale. The force generated by a single myosin head during the power stroke is about 5 to 10 piconewtons, equally tiny. But remember, in a single thick filament, there are about 600 myosin heads, and in a sarcomere, there are about 1,500 thick filaments. If even a fraction of these heads are performing power strokes at any given moment, the forces add up rapidly. After the power stroke, the myosin head is still attached to actin, now in its low-energy bent conformation, and ADP is still bound to the myosin head. The ADP is released shortly after the power stroke, which brings us back to where we started, myosin bound to actin with no nucleotide attached. The cycle is ready to begin again when another ATP molecule binds. This might seem like a slow process, but it's remarkably fast. Under optimal conditions, a single myosin head can complete the entire cycle from ATP binding through power stroke and back to ATP binding in about 50 milliseconds. That's 20 cycles per second. And because the hundreds of myosin heads on a thick filament are cycling asynchronously, out of phase with each other, there are always some heads bound to actin, pulling, while others are detached and repositioning. This ensures continuous force generation as long as ATP and calcium are available. The asynchronous cycling of myosin heads is critical for smooth force production. If all the myosin heads cycled in synchrony, binding and releasing simultaneously, the force would fluctuate dramatically, pulsing on and off. The rate of cross-bridge cycling, and therefore the speed of muscle contraction, depends on several factors. The most important is the myosin isoform, the specific version of the myosin heavy chain protein expressed in the muscle fiber. Different myosin isoforms have different ATPase activities, meaning they hydrolyze ATP and complete the cross-bridge cycle at different rates. Fast-twitch muscle fibers express myosin isoforms with high ATPase activity, allowing them to contract quickly. Slow-twitch fibers express myosin isoforms with lower ATPase activity, resulting in slower but more economical contraction. Temperature also affects cross-bridge cycling rate. Like most biochemical reactions, the cross-bridge cycle proceeds faster at higher temperatures. This is why muscles contract more slowly when cold, and why warming up before intense activity improves performance. Each 10 degrees Celsius increase in muscle temperature roughly doubles the rate of cross-bridge cycling, at least until temperatures get high enough to denature proteins. The concentration of ATP also matters, though under normal physiological conditions, ATP concentration is maintained at relatively constant levels. It's only in pathological conditions, or in rigor mortis after death, when ATP production ceases, that ATP depletion becomes a limiting factor. When ATP is absent, myosin heads bind to actin and cannot detach, producing the stiffness characteristic of rigor mortis. The availability of calcium is the primary physiological regulator of cross-bridge cycling. Without calcium, tropomyosin blocks the myosin binding sites on actin, preventing cross-bridge formation. No matter how much ATP is available, no matter how eager the myosin heads are to bind, they simply cannot access actin. When calcium binds to troponin C and tropomyosin shifts out of the way, the myosin binding sites are exposed and cross-bridge cycling can proceed. This makes calcium the on-off switch for muscle contraction. The amount of force a muscle generates depends not only on whether cross bridges are cycling, but also on how many cross bridges are cycling simultaneously. This, in turn, depends on the degree of overlap between thick and thin filaments. When a sarcomere is at its optimal length, about 2 to 2.2 micrometers, there is maximal overlap between the thick and thin filaments. Every myosin head has the opportunity to interact with actin, and force production is maximal. If the sarcomere is stretched beyond this optimal length, the overlap between thick and thin filaments decreases. Fewer myosin heads can reach actin, and force production declines. At very long sarcomere lengths, beyond about 3.6 micrometers, there is no overlap at all, and the muscle cannot generate active force. This is why a muscle stretched to extreme lengths feels weak. There simply aren't enough cross bridges forming to generate meaningful force. Conversely, if the sarcomere is compressed to very short lengths, below about 1.7 micrometers, force production also declines. At these short lengths, the thin filaments from opposite sides of the sarcomere begin to overlap in the center, interfering with each other. Additionally, the thick filaments may run into the Z-discs, causing mechanical interference. The result is that even though there is substantial overlap between thick and thin filaments, the geometry is wrong for optimal force production. This length-tension relationship has important functional implications. Muscles attached to the skeleton operate at lengths close to their optimal sarcomere length, where force production is maximal. This is not accidental. The arrangement of muscles and bones positions the muscles so that, over their normal operating range, sarcomeres remain close to optimal length. However, at the extremes of joint motion, when muscles are either fully stretched or fully shortened, force production declines, which is why you feel weaker at the extreme ends of a joint's range of motion. The sliding filament mechanism also explains why muscles can only pull, never push. The myosin heads pull the thin filaments toward the center of the sarcomere. There is no mechanism for them to push the filaments outward. This unidirectional force generation is a fundamental constraint on muscle function and explains why muscles must be arranged in opposing pairs or groups to produce bidirectional movement at joints. During a contraction, the thin filaments slide toward the center of the sarcomere from both sides. The Z discs are pulled closer together, the I band narrows, and the H zone narrows or disappears entirely. The A band, corresponding to the length of the thick filaments, remains constant in width because the thick filaments themselves don't change length. This observation, made by the early researchers studying muscle under the microscope, was one of the key pieces of evidence supporting the sliding filament theory. The maximum distance a sarcomere can shorten is about 50%, from roughly 2.5 micrometers at rest to about 1.2 to 1.3 micrometers at maximal contraction. If a muscle fiber is 10 centimeters long at rest and contains about 40,000 sarcomeres in series, then each sarcomere shortening by 1.2 micrometers results in the muscle shortening by about 4.8 centimeters, or roughly 48% of its resting length. This explains why muscles can produce substantial changes in length, enough to move joints through large ranges of motion. The velocity of shortening, how fast the muscle contracts, also affects force production. This relationship, called the force-velocity relationship, is one of the fundamental properties of muscle. When a muscle contracts against a very light load, it can shorten very quickly, but the force it generates is low. As the load increases, the velocity of shortening decreases, and the force increases. At some point, the load is so heavy that the muscle cannot shorten at all, and it produces what's called an isometric contraction, generating maximum force with no change in length. The force-velocity relationship exists because of the mechanics of cross-bridge cycling. When the muscle is shortening rapidly, the myosin heads don't have much time in contact with actin during each cycle. They bind, perform a quick power stroke, and release before they can generate maximum force. Additionally, the rapid sliding of the filaments means that some myosin heads are forcibly detached before completing their power stroke, reducing efficiency. As the load increases and shortening velocity decreases, the myosin heads have more time in contact with actin, can complete fuller power strokes, and more of them are attached at any given moment, all of which increases force production. Interestingly, muscle can also generate force while lengthening during what's called an eccentric contraction. This occurs when an external force stretches the muscle while it's trying to contract, such as when you lower a heavy weight slowly or walk downhill. During eccentric contractions, the cross bridges are forcibly stretched and detached by the external force. This can actually generate more force than isometric or concentric contractions, but it also causes more muscle damage, particularly if the muscle isn't accustomed to eccentric loading. This is why walking downhill often produces more muscle soreness than walking uphill, even though it feels easier at the time. The efficiency of converting chemical energy to mechanical work during cross-bridge cycling is actually quite high by biological standards, approaching 50% under optimal conditions. This means that about half the energy from ATP hydrolysis is converted to mechanical work, with the other half dissipated as heat. This is far better than most human-made engines, where efficiencies are typically 20 to 30%. However, when you account for all the other ATP-consuming processes in muscle, including calcium pumping and maintaining ion gradients, overall muscle efficiency drops to about 20 to 25%. The cross-bridge cycle continues as long as two conditions are met. ATP is available, and calcium is present in sufficient concentration to keep the myosin binding sites on actin exposed. When either condition fails, contraction ceases. If calcium is pumped back into the sarcoplasmic reticulum and tropomyosin blocks the binding sites, myosin heads cannot attach to actin, and the muscle relaxes. If ATP is depleted, myosin heads bind to actin but cannot detach, producing the rigid state of rigor mortis. The discovery of the sliding filament mechanism and the elucidation of the cross-bridge cycle represent one of the great achievements of 20th century biology. These discoveries required the convergence of multiple technologies and approaches: light and electron microscopy to visualize muscle structure, biochemistry to identify and characterize the proteins involved, biophysics to measure forces and movements at the molecular scale, and physiology to understand how the system operates in living muscle. The result is a remarkably complete picture of how molecular motors convert chemical energy into mechanical force. Yet even this complete picture raises new questions. How does the electrical signal from a motor neuron trigger the release of calcium from the sarcoplasmic reticulum? How is the signal transmitted from the surface of the muscle fiber to the depths of the cell where the myofibrils reside? The answers to these questions lie in the process of excitation-contraction coupling, which we must now explore to understand how the nervous system commands muscle to contract. Skeletal muscle, for all its sophisticated molecular machinery, is fundamentally passive. Left to its own devices, it will not contract. The sarcomeres contain all the necessary proteins. The muscle fiber stores ample ATP and glycogen, but without a signal from the nervous system, the muscle remains relaxed. This signal arrives at a specialized synapse called the neuromuscular junction, a site where a motor neuron terminal comes into close contact with a muscle fiber. The events that unfold at this junction, transforming an electrical signal in a nerve into an electrical signal in muscle, represent one of the most studied examples of synaptic transmission in all of neuroscience. Motor neurons, the nerve cells that innervate skeletal muscle, have their cell bodies located in the ventral horn of the spinal cord or in the motor nuclei of cranial nerves in the brainstem. Their axons, sometimes extending for a meter or more in large humans, travel through peripheral nerves to reach their target muscles. As the motor axon approaches the muscle, it branches repeatedly, with each branch innervating a separate muscle fiber. A single motor neuron may innervate anywhere from a few muscle fibers to several thousand, depending on the muscle and its function. The motor neuron axon is myelinated for most of its length, wrapped in insulating myelin sheaths produced by Schwann cells. This myelination allows action potentials to propagate rapidly along the axon through saltatory conduction, jumping from one node of Ranvier to the next at speeds up to 120 meters per second in the largest motor axons. As the axon approaches the muscle fiber, It loses its myelin sheath and begins to branch into multiple terminal boutons, small swellings at the ends of the finest axon branches. Each terminal bouton forms a synapse with a single muscle fiber at a site called the motor end plate. This is a specialized region of the sarcolemma, typically located near the middle of the muscle fiber, where the muscle fiber membrane is folded into deep junctional folds. These folds increase the surface area of the muscle fiber membrane manyfold, packed with acetylcholine receptors and other proteins necessary for responding to the neurotransmitter signal. The space between the motor neuron terminal and the motor end plate is called the synaptic cleft, about 50 nanometers wide. This is much narrower than the extracellular space elsewhere, but it's still a gap that must be bridged by chemical signals. The motor neuron terminal contains thousands of small membrane-bound vesicles, each about 50 nanometers in diameter, filled with acetylcholine, the neurotransmitter used at the neuromuscular junction. Each synaptic vesicle contains about 5,000 to 10,000 molecules of acetylcholine. The vesicles are clustered near specialized release sites on the presynaptic membrane, areas where the machinery for vesicle fusion and neurotransmitter release is concentrated. Some vesicles are docked at these release sites, primed and ready to fuse with the membrane at a moment's notice. Others are held in reserve nearby, ready to move to the release sites as needed. When an action potential arrives at the motor neuron terminal, it causes voltage-gated calcium channels in the presynaptic membrane to open. Calcium ions rush into the terminal from the synaptic cleft, where calcium concentration is about 2 millimolar, into the terminal, where calcium concentration is normally maintained at very low levels, about 0.1 micromolar. This thousandfold increase in calcium concentration triggers the fusion of synaptic vesicles with the presynaptic membrane. The mechanism by which calcium triggers vesicle fusion is complex and involves multiple proteins. Synaptotagmin, a calcium-sensing protein on the vesicle membrane, undergoes a conformational change when it binds calcium. This change allows synaptotagmin to interact with snare proteins, a complex of proteins that brings the vesicle membrane and the plasma membrane into close apposition. When the membranes are brought close enough together, they fuse, and the vesicle opens, releasing its contents into the synaptic cleft. This process, from calcium influx to neurotransmitter release, occurs with remarkable speed within 200 microseconds of the calcium channel's opening. The rapid release is essential for the speed and precision of neuromuscular transmission. A single action potential arriving at the motor neuron terminal typically triggers the release of 50 to 300 synaptic vesicles simultaneously, dumping several 100,000 molecules of acetylcholine into the synaptic cleft. The acetylcholine molecules diffuse across the narrow synaptic cleft in about 0.5 milliseconds, quickly reaching the junctional folds of the motor end plate. There, they encounter nicotinic acetylcholine receptors, ligand-gated ion channels that open when acetylcholine binds. Each receptor is composed of 5 protein subunits arranged in a circle, forming a central pore through the membrane. The receptor has 2 binding sites for acetylcholine, and both must be occupied for the channel to open efficiently. When acetylcholine binds and the channel opens, it allows sodium and potassium ions to flow through. Because sodium concentration is much higher outside the cell than inside, and because the electrical potential across the membrane favors sodium entry, sodium flows into the cell much more than potassium flows out. This influx of positive charges depolarizes the muscle fiber membrane, making the inside less negative relative to the outside. The depolarization at the motor end plate is called an end plate potential, or EPP. Unlike action potentials, which are all or none events, the end plate potential is a graded response. Its amplitude depends on how much acetylcholine is released and how many receptors are activated. However, under normal conditions, the amount of acetylcholine released by a single action potential in the motor neuron is far more than necessary to trigger an action potential in the muscle fiber. This provides a large safety factor, ensuring reliable transmission even under conditions that might compromise neuromuscular function. A typical end plate potential might depolarize the motor end plate by 70 to 80 millivolts from the resting potential of about negative 90 millivolts to around negative 10 or negative 20 millivolts. This is well above the threshold for triggering an action potential in the surrounding sarcolemma, which is typically around negative 55 millivolts. So the end plate potential doesn't just reach threshold, it overshoots it substantially, ensuring that an action potential is triggered. The action potential is initiated in the sarcolemma immediately adjacent to the motor end plate, where there is a high density of voltage-gated sodium channels. When the end plate potential depolarizes this region above threshold, the sodium channels open, allowing a regenerative influx of sodium that produces the upstroke of the action potential. The action potential then propagates away from the motor end plate in both directions along the muscle fiber, traveling at about 3 to 5 meters per second. This is much slower than action potential propagation in myelinated axons, which can reach speeds of 120 meters per second. The difference is that the sarcolemma is not myelinated, so the action potential must propagate through continuous local circuit currents rather than saltatory conduction. Nevertheless, 3 to 5 meters per second is fast enough that the action potential reaches the ends of even the longest muscle fibers within milliseconds of being initiated at the motor end plate. Meanwhile, back at the synaptic cleft, the acetylcholine released into the cleft doesn't remain active for long. The synaptic cleft contains high concentrations of an enzyme called acetylcholinesterase, which rapidly breaks down acetylcholine into acetate and choline. This enzyme is extraordinarily efficient, with each molecule capable of hydrolyzing thousands of acetylcholine molecules per second. Within about 1 to 2 milliseconds of release, virtually all the acetylcholine in the synaptic cleft has been destroyed. This rapid inactivation of acetylcholine is essential for allowing the muscle to relax and for enabling rapid, repeated contractions. If acetylcholine remained active in the cleft, the muscle would remain depolarized and contracted continuously. The choline produced by acetylcholinesterase is transported back into the motor neuron terminal, where it is used to synthesize new acetylcholine, conserving this essential molecule. The importance of acetylcholinesterase becomes apparent in cases where it is inhibited. Many organophosphate insecticides and nerve agents work by irreversibly inhibiting acetylcholinesterase, causing acetylcholine to accumulate in the synaptic cleft. The resulting continuous stimulation of muscle fibers causes persistent contraction, fasciculations, and eventually paralysis as the muscle becomes incapable of responding to further stimulation. This is why these compounds are so toxic. On the other hand, some drugs that reversibly inhibit acetylcholinesterase can be therapeutic. In myasthenia gravis, an autoimmune disease where antibodies destroy acetylcholine receptors at the motor end plate, drugs like pyridostigmine can prolong the action of acetylcholine, compensating for the reduced number of receptors and improving muscle strength. After the end plate potential triggers an action potential, the muscle fiber's response depends on what happens next. The action potential itself doesn't directly cause contraction. It's just an electrical signal propagating along the sarcolemma. For contraction to occur, this electrical signal must somehow reach the myofibrils deep within the muscle fiber and trigger the release of calcium from the sarcoplasmic reticulum. The tubules are invaginations of the sarcolemma that plunge deep into the muscle fiber, running perpendicular to the myofibrils. They are positioned at specific locations along the sarcomere, at the junctions between the A bands and I bands in mammalian skeletal muscle. This positioning is not arbitrary. It places the tubules precisely where they need to be to activate the contractile apparatus most effectively. When the action potential propagates along the sarcolemma, it also propagates down into the tubules, reaching deep into the muscle fiber within milliseconds. The tubule membrane contains the same voltage-gated ion channels as the surface sarcolemma, so the action potential is actively regenerated as it travels down the tubule, ensuring that the signal doesn't decay with distance. At the end of each tubule are terminal cisternae of the sarcoplasmic reticulum, the calcium storage compartments we discussed earlier. The tubule membrane and the terminal cisternae membrane come into very close contact, separated by only about 15 nanometers. At this junction, there are protein complexes that physically link the two membranes and allow the electrical signal in the tubule to trigger calcium release from the sarcoplasmic reticulum. This process is called excitation-contraction coupling, and it is the subject of our next chapter. The neuromuscular junction, despite its small size and seemingly simple function, is actually a marvel of biological engineering. It converts an electrical signal in a nerve into a chemical signal, which then triggers an electrical signal in muscle, all within a few milliseconds. The design includes multiple safety factors and redundancies to ensure reliable transmission. The amount of acetylcholine released is far more than the minimum required. The number of acetylcholine receptors at the motor end plate is far more than necessary. These safety factors ensure that neuromuscular transmission is extraordinarily reliable under normal conditions. However, the neuromuscular junction is also a point of vulnerability. Numerous diseases and toxins target this junction. Myasthenia gravis, as mentioned, destroys acetylcholine receptors. Lambert-Eaton syndrome interferes with acetylcholine release from the motor neuron terminal. Botulinum toxin, one of the most potent toxins known, blocks acetylcholine release, causing paralysis. Curare and related compounds block acetylcholine receptors and are used as muscle relaxants during surgery. Understanding the neuromuscular junction has been essential for understanding these conditions and developing treatments. The neuromuscular junction also has the capacity to adapt and change. With training, particularly strength training, the morphology of the neuromuscular junction can change with increased surface area of the motor end plate and increased density of acetylcholine receptors. These changes may contribute to the neural adaptations that accompany strength training, improving the efficiency of neuromuscular transmission. Conversely, with disuse or denervation, the neuromuscular junction can degenerate, with muscle fibers losing their innervation and becoming atrophic. From signal to synapse, from neurotransmitter to receptor, from end plate potential to action potential, the neuromuscular junction orchestrates the first critical steps in muscle activation. But the action potential, spreading across the sarcolemma and down the tubules is still just an electrical signal. The actual mechanical contraction requires that this signal be coupled to the release of calcium, the trigger that initiates cross-bridge cycling. And so we turn now to the remarkable mechanism that links excitation to contraction, that transforms electricity into chemistry into motion. The gap between electrical excitation and mechanical contraction might seem small, a simple matter of connecting one event to the next. But in reality, this gap represents one of the most elegant and sophisticated signaling systems in the body. The action potential traveling along the sarcolemma and down the tubules must somehow trigger the release of calcium from the sarcoplasmic reticulum, and this calcium must then diffuse to the myofibrils and bind to troponin to initiate contraction. All of this must happen quickly, within just a few milliseconds, to allow for the rapid contractions that skeletal muscle is capable of producing. The key to this rapid signaling is the close physical relationship between the tubule membrane and the sarcoplasmic reticulum membrane at structures called triads. At each triad, a tubule is sandwiched between two terminal cisternae of the sarcoplasmic reticulum, with the membranes separated by only about 15 nanometers. Spanning this narrow gap are protein complexes that physically link the two membranes and provide the molecular machinery for excitation-contraction coupling. This calcium diffuses rapidly to the thin filaments, a distance of only about 1 micrometer from the terminal cisternae to the nearest myofibril. Calcium binds to troponin C, the calcium-sensing subunit of the troponin complex. As we discussed earlier, this binding triggers a conformational change in the troponin complex, causing tropomyosin to shift position and expose the myosin binding sites on actin. Cross-bridge cycling begins, and the muscle contracts. The entire process, from action potential depolarizing the tubule to the onset of force development, takes only about 10 to 15 milliseconds in fast-twitch skeletal muscle. This is remarkably fast for a biological signaling system. Consider that the action potential itself lasts only about 2 milliseconds. The excitation-contraction coupling takes another 5 milliseconds, and force begins to rise almost immediately thereafter. The speed of this system allows skeletal muscle to generate rapid, precise movements. But contraction is only half the story. For the muscle to relax and be ready for the next contraction, the calcium must be removed from the sarcoplasm, returned to the sarcoplasmic reticulum. This is the job of the sarcoplasmic reticulum calcium ATPase pumps, or SERCA pumps. These molecular pumps sit in the membrane of the sarcoplasmic reticulum and actively transport calcium from the sarcoplasm back into the reticulum, using ATP as an energy source. Each SERCA pump transports 2 calcium ions per ATP molecule hydrolyzed. This might not sound very efficient, but the pumps are numerous, and they're extraordinarily effective at lowering calcium concentration. They can reduce the sarcoplasmic calcium concentration back to resting levels within 30 milliseconds or so after calcium release stops. The speed of this calcium removal is actually one of the factors limiting how fast muscle can contract and relax repeatedly. Fast-twitch muscles have higher densities of SERCA pumps than slow-twitch muscles, allowing them to relax more quickly. Inside the sarcoplasmic reticulum, calcium doesn't just float freely. It's bound to proteins called calsequestrin and calreticulin, which buffer the free calcium concentration and allow the sarcoplasmic reticulum to store much more calcium than would otherwise be possible. Without these buffer proteins, the calcium concentration inside the reticulum would be so high that it would precipitate with phosphate, forming calcium phosphate crystals. The buffer proteins keep the calcium in solution while still allowing rapid release when the ryanodine receptors open. The amount of calcium released during a single action potential doesn't activate all the troponin in the muscle fiber. Rather, only about 10 to 20% of troponin molecules bind calcium during a single twitch. This provides a mechanism for grading force production if action potentials arrive repeatedly at high frequency before the muscle has fully relaxed. Calcium doesn't have time to be pumped back into the sarcoplasmic reticulum between action potentials. The sarcoplasmic calcium concentration remains elevated. More troponin molecules bind calcium, more cross bridges form, and force increases. This phenomenon, called summation, allows the muscle to produce graded force output depending on the frequency of motor neuron firing. At very high stimulation frequencies, above about 50 to 100 action potentials per second, depending on the muscle, the calcium concentration remains high enough that the muscle doesn't relax at all between stimuli. Instead, it produces a sustained, smooth contraction called tetanus. This is actually the normal mode of muscle activation during most voluntary movements. When you reach for an object or maintain posture, your motor neurons are firing at frequencies that produce tetanic contractions, not individual twitches. The term tetanus in this context should not be confused with the disease tetanus caused by the bacterium Clostridium tetani, though the disease is named for the sustained muscle contractions it causes. In the disease, the bacterial toxin interferes with inhibitory synapses in the spinal cord, leading to excessive motor neuron activity and prolonged, painful muscle contractions. The regulation of calcium in muscle is so critical that disruptions in this system can be fatal. Malignant hyperthermia is a genetic condition where mutated ryanodine receptors become abnormally sensitive to certain anesthetic drugs, particularly halothane. When affected, individuals are exposed to these drugs. The ryanodine receptors open uncontrollably, flooding the sarcoplasm with calcium. The muscle contracts continuously, generating enormous amounts of heat, which can raise body temperature to dangerous levels, sometimes above 40 degrees Celsius. The continuous muscle contraction also consumes ATP faster than it can be regenerated, leading to cellular damage and the release of muscle proteins into the bloodstream. The steepness of this relationship has functional implications. It means that small changes in the frequency of action potentials, which produce small changes in average calcium concentration, can produce relatively large changes in force output. This provides fine control over force production. It also means that the muscle operates on a fairly narrow range of calcium concentrations where force is most sensitive to calcium changes, optimizing the muscle's responsiveness. In smooth muscle, calcium's role is similar in principle, but different in detail. Smooth muscle also uses calcium as the trigger for contraction, but the mechanism differs from skeletal muscle. As mentioned earlier, smooth muscle lacks troponin. Instead, calcium binds to calmodulin, a small calcium-binding protein found in all cell types. The calcium calmodulin complex then activates an enzyme called myosin light chain kinase, which phosphorylates the regulatory light chains on myosin. Only when these light chains are phosphorylated can myosin bind to actin and generate force. This phosphorylation-based regulation is slower than the troponin-tropomyosin system of skeletal muscle. It takes several seconds for myosin light chain kinase to phosphorylate enough myosin to produce maximal force, compared to the milliseconds required in skeletal muscle. However, the smooth muscle system has an advantage. It can maintain force with much less energy expenditure. Once the myosin is phosphorylated and the cross bridges are attached, a phosphatase enzyme slowly removes the phosphate groups. If this happens while the myosin is still attached to actin, the cross bridge can remain attached without cycling, a state called a latch bridge. These latch bridges maintain tension without consuming ATP, allowing smooth muscle to sustain contraction for hours or even days with minimal energy cost. This is why smooth muscle in the walls of blood vessels can maintain vascular tone continuously, why the smooth muscle of the uterus can sustain contractions during labor for hours, and why the smooth muscle of the digestive tract can maintain segmental contractions that help mix food. The energy cost of these sustained contractions would be prohibitive if smooth muscle used the same mechanism as skeletal muscle. In cardiac muscle, the excitation contraction coupling mechanism resembles that of skeletal muscle, with tubules and sarcoplasmic reticulum working together but with important differences. As mentioned, cardiac muscle uses calcium-induced calcium release rather than mechanical coupling. The tubules are positioned differently at the Z discs rather than at the AI junctions. The sarcoplasmic reticulum is less extensive, and cardiac muscle is more dependent on calcium entering from outside the cell through the L-type calcium channels. This makes cardiac muscle more sensitive to extracellular calcium concentration than skeletal muscle. Cardiac muscle also has mechanisms to prevent excessive calcium entry, which could lead to calcium overload and cell damage. The sodium-calcium exchanger in the sarcolemma uses the energy from sodium moving into the cell down its concentration gradient to pump calcium out of the cell. This exchanger works together with the circa pumps to regulate calcium concentration. Disruption of this delicate balance, as occurs in heart failure, can lead to abnormal calcium handling, arrhythmias, and impaired contractility. The dependence of cardiac muscle on extracellular calcium is exploited therapeutically. Calcium channel blockers, drugs that block L-type calcium channels, are used to treat hypertension and certain arrhythmias by reducing calcium entry into cardiac muscle cells, decreasing the force of contraction and slowing the heart rate. Conversely, drugs that increase intracellular calcium, such as cardiac glycosides like digoxin, increase the force of contraction and are used to treat heart failure, though with careful monitoring due to the narrow therapeutic window. The story of excitation-contraction coupling is a story of molecular precision and speed. From voltage sensors to calcium channels, from calcium-binding proteins to molecular pumps, every component has been refined by evolution to respond within milliseconds and to do so reliably, billions of times over a lifetime. The fact that this system works so well, that we can voluntarily contract and relax our muscles with such precision and speed, is a testament to the sophistication of biological design. Yet even this remarkable signaling system cannot produce graded, controlled force output on its own. A single muscle fiber responds to an action potential with a twitch, a brief contraction followed by relaxation. To produce sustained, variable force output, the nervous system must control not just whether muscle fibers contract, but how many contract and how often. A single muscle fiber is a binary device. Either it receives an action potential and contracts, or it doesn't receive a signal and remains relaxed. There's no halfway. The fiber either generates force or it doesn't. This all or none property of muscle fiber contraction poses a problem. How does the nervous system produce the graduated, precisely controlled forces necessary for everything from lifting a pencil to lifting a barbell, from whispering to shouting, from tiptoeing to stomping? The answer lies in the organization of motor units and the mechanisms by which the nervous system recruits these units and controls their firing rates. A motor unit consists of a single motor neuron and all the muscle fibers it innervates. When the motor neuron fires an action potential, all the muscle fibers in that motor unit contract together. The motor unit is thus the smallest functional element of muscle control. You cannot activate individual muscle fibers within a motor unit independently. They're all controlled by the same neuron, so they all contract as a group. The size of motor units varies dramatically across different muscles and reflects the functional demands placed on those muscles. In muscles that must produce very fine, precise movements, motor units are small, sometimes containing as few as 5 to 10 muscle fibers. The extraocular muscles that control eye movements, for instance, have motor units averaging about 10 fibers per motor neuron. This allows for extremely precise control of eye position, essential for tracking objects and maintaining clear vision. In contrast, muscles that produce large forces but don't require fine control have much larger motor units. The gastrocnemius muscle in the calf, used for powerful plantar flexion when jumping or running, has motor units containing 1,000 to 2,000 muscle fibers per motor neuron. The medial gastrocnemius alone contains about 1,000 motor units, which means this single muscle contains roughly 1 to 2 million muscle fibers. Large motor units allow a small number of motor neurons to control a large amount of muscle tissue, which is efficient for generating force, though at the cost of fine control. The relationship between motor unit size and function is sometimes called the size principle, though this term is more commonly used to describe the order in which motor units are recruited, which we'll discuss shortly. Muscles requiring fine control have more motor units and smaller motor units. Muscles requiring large forces have fewer, larger motor units. This organization reflects a fundamental trade-off in motor control: precision versus power. When you decide to contract a muscle, your motor cortex sends signals down to the motor neurons in the spinal cord, and those motor neurons begin to fire action potentials. The nervous system has two primary mechanisms for controlling the force produced by the muscle. The first is recruitment, activating more motor units. The second is rate coding, increasing the frequency at which each motor unit fires. Recruitment is straightforward in principle. If you want to produce a small force, you activate a few motor units. If you want more force, you activate more motor units. The relationship between the number of active motor units and the force produced is roughly linear, though with some complications we'll explore. For many muscles, at maximal voluntary contraction, essentially all motor units are active, though the extent to which all motor units can be activated varies across muscles and individuals. The order in which motor units are recruited is not random. This brings us to the Henneman size principle, one of the fundamental principles of motor control discovered by Elwood Henneman in the 1960 seconds. The size principle states that motor units are recruited in order of increasing size, from smallest to largest. Small motor units, with small motor neurons and few muscle fibers, are recruited first. As force requirements increase, progressively larger motor units are recruited. Why this orderly recruitment? The explanation lies in the properties of motor neurons themselves. Small motor neurons have smaller cell bodies, fewer dendrites, and higher input resistance. This makes them more excitable. A given synaptic input produces a larger depolarization in a small motor neuron than in a large motor neuron, so the small motor neuron reaches threshold first. As the descending drive from the brain increases, small motor neurons fire first, then progressively larger motor neurons are recruited as the drive increases further. This orderly recruitment has important functional consequences. The muscle fibers in small motor units tend to be slow twitch, fatigue-resistant fibers. They generate small forces, but can maintain those forces for long periods. The muscle fibers in large motor units tend to be fast twitch, fatigable fibers. They generate large forces, but fatigue quickly. If a second action potential arrives before the muscle has fully relaxed from the first twitch, the forces sum. This is called summation. The calcium concentration in the sarcoplasm doesn't return to baseline between action potentials, so more troponin molecules remain bound to calcium, more cross bridges remain attached, and force is higher than during a single twitch. As firing frequency increases further, summation becomes more pronounced, until, at high frequencies, typically 50 to 100 hertzes for most muscles, the force plateaus at a maximum. This sustained, maximal contraction is called tetanus. The relationship between firing frequency and force is sigmoidal. At low frequencies, small increases in frequency produce small increases in force. In the middle range, force increases steeply with frequency. At high frequencies, further increases produce little additional force. This sigmoidal relationship is functionally useful. It means the nervous system can make large changes in force with relatively modest changes in firing frequency, providing good force resolution in the range where it's most needed. The combination of recruitment and rate coding gives the nervous system flexible control over force production. For very small forces, a few small motor units fire at low rates. As force requirements increase, firing rates increase, and more motor units are recruited. At maximal force, all motor units are active and firing at high frequencies. The relative contribution of recruitment versus rate coding varies across muscles. In muscles with many small motor units, recruitment is the primary mechanism, with rate coding playing a secondary role. In muscles with fewer, larger motor units, rate coding becomes more important. The force produced by a single motor unit depends not only on the number of muscle fibers it contains, but also on the fiber type. Motor units with slow twitch fibers produce lower forces, typically a few hundred millinewtons, but can sustain those forces for long periods without fatigue. Motor units with fast twitch fibers produce higher forces, sometimes several newtons, but fatigue within a few minutes or even seconds of continuous activity. The variation in motor unit force can span a 100-fold range within a single muscle. This variation allows for both fine force control and large maximum force. When small, weak motor units are recruited first, small adjustments in force can be made by recruiting just a few more motor units or modestly changing firing rates. As stronger motor units are recruited, the same number of additional motor units adds more force, allowing for rapid force increases when needed. This is why it's easy to make small adjustments when holding a light object, but harder to make fine adjustments when holding something heavy. The stronger motor units required for the heavy load have less fine force resolution. Motor units are not uniformly distributed throughout a muscle. Instead, the muscle fibers belonging to a single motor unit are scattered across a territory that may encompass 10 to 30% of the muscle's cross-sectional area. This distributed arrangement means that when a motor unit is active, it produces force across a relatively wide region of the muscle rather than in a single localized spot. This helps produce smooth, even tension throughout the muscle and may help prevent local fatigue. Interestingly, muscle fibers from different motor units intermingle quite extensively. In a small region of muscle, you might find fibers from dozens of different motor units interleaved with each other. This checkerboard pattern is thought to result from the competitive interactions during development as motor neurons extend branches to muscle fibers and compete for territory. The pattern has functional advantages, ensuring that even when only a few motor units are active, force is distributed relatively evenly across the muscle. The nervous system's control of motor units is not entirely conscious. When you decide to pick up a cup of coffee, you're not explicitly thinking about which motor units to recruit or how fast they should fire. That control occurs at lower levels of the motor system, in the spinal cord and brainstem, implementing motor programs that have been learned through experience. The motor cortex specifies the goal, pick up the cup, and lower motor centers work out the details of which muscles to activate, which motor units to recruit, and at what rates they should fire. This hierarchical control allows for both flexibility and efficiency. High-level motor areas can specify goals and general strategies without getting bogged down in the details of motor unit control. Low-level motor areas implement these strategies using learned patterns that can be executed quickly and automatically. This is why you can pick up a cup without consciously thinking about contracting your biceps and triceps in precisely the right proportions. The motor program handles it. Learning a new motor skill involves, in part, learning new motor unit recruitment patterns. When you first try to play a musical instrument or learn a new sport, your motor unit control is crude. You activate too many motor units, co-contract muscles that shouldn't be active, and struggle to produce smooth, graded forces. With practice, motor unit control becomes more refined. You learn to recruit only the necessary motor units, to activate them at appropriate rates, and to coordinate their activity with other muscles. This refinement of motor control is one aspect of motor learning. The motor unit is also the site where motor neuron diseases manifest their effects. In amyotrophic lateral sclerosis, or ALS, motor neurons progressively degenerate and die. As each motor neuron is lost, the muscle fibers in its motor unit become denervated and atrophy. Neighboring motor neurons may sprout new branches and reinnervate some of the denervated fibers, temporarily compensating for the loss. But as more motor neurons die, eventually there aren't enough motor units left to produce functional movement, and paralysis ensues. In contrast, in myasthenia gravis, the motor units remain intact, but neuromuscular transmission fails due to autoimmune destruction of acetylcholine receptors. The motor neurons fire normally, but the muscle fibers respond weakly or not at all. This produces a different pattern of weakness, one that worsens with repeated muscle use as the already compromised neuromuscular junctions become further impaired by repeated transmitter release. The concept of the motor unit also helps explain muscle fatigue. During sustained or repeated contraction, motor units fatigue at different rates depending on their fiber type composition. Slow twitch motor units, recruited early, can sustain activity for long periods. Fast twitch motor units, recruited later, fatigue quickly. As fast twitch motor units fatigue and drop out, the nervous system must recruit additional motor units to maintain force. Eventually, when many motor units have fatigued, The nervous system may not be able to recruit enough active motor units to produce the required force, and task failure occurs. Interestingly, the nervous system appears to have mechanisms to sense motor unit fatigue and to adjust recruitment strategies accordingly. If a motor unit is fatigued, the nervous system may de-recruit it and recruit a fresh motor unit, allowing the fatigued unit time to recover. This rotation of motor units may help prolong endurance during sustained tasks, though the extent to which this occurs and the mechanisms involved are still being studied. The motor unit, then, is far more than just an anatomical detail. It's a fundamental functional element of motor control, the unit through which the nervous system commands muscle. The properties of motor units, their size, fiber type composition, and recruitment order all reflect evolutionary optimization for the diverse demands of movement. From the delicate control of eye movements to the powerful contractions of locomotion, motor units provide the interface between neural intention and muscular action. But motor units, for all their importance, represent only one level of functional organization. The muscle fibers themselves come in different varieties, each optimized for different mechanical and metabolic demands, and it's to these different fiber types that we now turn our attention. Not all muscle fibers are created equal. Within a single muscle, you'll find fibers with dramatically different properties. Some contract slowly, but resist fatigue for hours. Others contract rapidly, but exhaust themselves within seconds. These differences aren't accidents of development or random variation. They reflect specialization, the result of evolutionary pressure to optimize different fibers for different tasks. Understanding these fiber types and their properties is essential to understanding how muscles function and how they adapt to training. The classic classification of skeletal muscle fibers divides them into three main types based on their contractile and metabolic properties. Type 1 fibers, also called slow twitch or slow oxidative fibers, contract relatively slowly but are highly resistant to fatigue. Type IEA fibers, called fast oxidative glycolytic fibers, contract quickly and have moderate fatigue resistance. Type 2X fibers, sometimes called type IIB in older literature, though true. Type IIB fibers are rare in humans, are fast-twitch glycolytic fibers that contract very rapidly but fatigue quickly. These fiber types differ in almost every measurable property. Let's start with the myosin isoforms, the most fundamental difference. Type 1 fibers express a slow myosin heavy chain isoform with relatively low ATPase activity. When this myosin hydrolyzes ATP and goes through the cross-bridge cycle, it does so slowly, taking about 200 milliseconds to complete a cycle. Type 2A and Type 2X fibers express different fast myosin heavy chain isoforms, each with progressively higher ATPase activity. Type 2A myosin cycles faster than Type 1, and Type 2X cycles faster still. This allows Type 2X fibers to reach peak tension in as little as 30 to 40 milliseconds. The trade-off is efficiency. The faster myosin isoforms consume more ATP per unit of force produced, which partly explains why fast-twitch fibers fatigue more quickly. The metabolic properties of these fiber types differ as dramatically as their contractile properties. Type 1 fibers are optimized for aerobic metabolism. They're packed with mitochondria, which may occupy 25% or more of the fiber's volume. They have high concentrations of aerobic enzymes, particularly those involved in the citric acid cycle and electron transport chain. They have dense capillary networks surrounding them, typically 3 to 4 capillaries per fiber, ensuring excellent oxygen delivery. Type 1 fibers also contain high concentrations of myoglobin, an oxygen binding protein similar to hemoglobin. Myoglobin gives these fibers their red color and serves as an oxygen buffer, storing oxygen that can be used when metabolic demand temporarily exceeds oxygen delivery. The combination of abundant mitochondria, high enzyme levels, dense capillaries, and myoglobin makes type 1 fibers superbly equipped for sustained aerobic work. Type 2X fibers, at the opposite extreme, have relatively few mitochondria, perhaps occupying only 2 to 5% of fiber volume. They have lower concentrations of aerobic enzymes and less dense capillary networks, often only 1 capillary per fiber. They contain little myoglobin and appear pale or white in comparison to type 1 fibers. What they lack in aerobic capacity, they make up for in glycolytic capacity. Type 2X fibers have high concentrations of glycolytic enzymes, large glycogen stores, and can generate ATP rapidly through anaerobic glycolysis. Type IIA fibers are intermediate in most properties, possessing both substantial aerobic and glycolytic capacity. They have moderate mitochondrial content, perhaps 15% of fiber volume, good capillary density, and reasonable myoglobin content. They also have high glycolytic capacity. This makes them versatile, capable of producing rapid, forceful contractions like type 2X fibers, but with better endurance than pure glycolytic fibers. The force-generating capacity also differs between fiber types. Type 2X fibers can generate about twice as much force per cross-sectional area as type 1 fibers. This difference results partly from the faster myosin cycling rate, which allows more power strokes per second, and partly from differences in sarcomere organization and protein isoforms. The practical consequence is that for a given muscle size, a muscle with a higher proportion of fast-twitch fibers can generate more force. The fatigue resistance of these fiber types varies inversely with their force-producing capacity. Type 1 fibers can sustain contraction for hours without significant fatigue. During prolonged low-intensity exercise, type 1 fibers can maintain force production almost indefinitely, limited mainly by fuel availability and systemic factors like heat accumulation rather than local fatigue mechanisms. Type 2A fibers have good fatigue resistance, able to sustain activity for many minutes. They fatigue more quickly than type 1 fibers but recover more quickly as well, making them well suited for intermittent high-intensity work. Type 2X fibers fatigue rapidly, sometimes within 30 seconds of continuous maximal activation. Once fatigued, they require several minutes to recover substantially, and full recovery may take much longer. The mechanisms of fatigue differ between fiber types. In type 1 fibers, fatigue during prolonged activity is associated with glycogen depletion, accumulation of inorganic phosphate from ATP breakdown, and impaired calcium handling. In type 2X fibers, fatigue during intense activity is associated with metabolite accumulation, particularly hydrogen ions and inorganic phosphate, which interfere with cross-bridge cycling and calcium sensitivity. The proportion of different fiber types varies dramatically across muscles and across individuals. Postural muscles, like the soleus and the calf, are often 80 to 90% type 1 fibers, reflecting their role in sustained low-force contractions. Muscles used for rapid, powerful movements, like the gastrocnemius also in the calf, may be 50% or more fast-twitch fibers. Most muscles contain a mixture of fiber types, providing versatility for different tasks. Individual variation in fiber type composition is substantial and partly genetic. Some people are born with a higher proportion of slow twitch fibers, giving them natural endurance capacity. Others have more fast twitch fibers, providing an advantage in power and speed activities. Elite endurance athletes often have 70 to 80% or more type 1 fibers in their primary working muscles. Elite sprinters and power athletes often have 60 to 70% or more fast twitch fibers. These differences likely reflect both genetic predisposition and the effects of years of specialized training. Training can modify fiber properties, though it cannot completely transform one fiber type into another. Endurance training increases the oxidative capacity of all fiber types, increasing mitochondrial content, aerobic enzyme levels, and capillary density. This can make type 2A fibers more type I, like in their metabolic properties, though their myosin isoform and contractile speed don't fully convert. Sprint and strength training can increase the glycolytic capacity and size of fibers, particularly type 2A and 2IX fibers, making them more powerful, though not necessarily more fatigue resistant. One interesting finding is that type 2A fibers appear to be plastic, capable of shifting toward either more oxidative or more glycolytic phenotypes, depending on the training stimulus. With endurance training, they become more oxidative, expressing higher levels of mitochondrial proteins and taking on properties similar to type 1 fibers. With power training or detraining, they can shift toward a more glycolytic phenotype, becoming more similar to type 2X fibers. This flexibility makes type 2A fibers particularly responsive to training. There's also evidence that extreme inactivity or denervation can cause fibers to shift toward more glycolytic phenotypes. When a muscle is immobilized or denervated, even type 1 fibers can begin to lose oxidative capacity and shift toward a more type 2-like phenotype. This contributes to the rapid loss of endurance capacity that occurs with bed rest or limb immobilization. The distribution of fiber types within a muscle is not random. Fiber types are intermingled in a checkerboard pattern, as we discussed in the previous chapter, with fibers from different motor units intermixed. However, there can be regional variations. In some muscles, certain regions may have higher proportions of one fiber type, perhaps reflecting different functional demands on different parts of the muscle. Aging affects fiber type composition and properties. With aging, there's typically a loss of muscle mass, termed sarcopenia, and this loss affects type 2 fibers disproportionately. Fast twitch motor units may be lost through motor neuron death, and the remaining type 2 fibers may atrophy more than type 1 fibers. The result is that older muscles often have a higher proportion of type 1 fibers, though this reflects loss of type 2 fibers rather than conversion from one type to another. The loss of fast twitch fibers with aging has functional consequences. Power production declines more rapidly with aging than endurance capacity. Older adults often maintain good endurance for walking and other low-intensity activities, but struggle with rapid, powerful movements like rising from a chair, climbing stairs quickly, or catching themselves during a fall. This loss of power is a significant contributor to fall risk and loss of independence in elderly populations. There's also growing evidence that different fiber types may have different regenerative capacities and different susceptibilities to certain diseases. Type 2 fibers appear to regenerate more readily after injury, possibly because the satellite cells associated with them are more readily activated. Some muscular dystrophies preferentially affect certain fiber types, though the reasons for this selectivity are not fully understood. Nutritional factors can also influence fiber type properties. Adequate protein intake is essential for maintaining muscle mass in all fiber types, but the optimal protein intake may differ for endurance versus power athletes. Similarly, carbohydrate availability affects glycogen stores more in fast twitch fibers, which rely more heavily on glycolytic metabolism. Endurance training in a low carbohydrate state can enhance fat oxidation capacity, particularly in type 1 fibers, though potentially at the cost of reduced high intensity performance. The fiber type composition of a muscle also influences its response to hormones and other systemic factors. Fast twitch fibers appear to be more responsive to anabolic hormones like testosterone and growth hormone, which may partly explain why males with higher testosterone levels typically have larger muscles with a higher proportion of fast twitch fibers. Thyroid hormones also affect fiber type, with hypothyroidism favoring a shift towards slower, more type I-like properties, and hyperthyroidism favoring faster properties. In disease states, fiber type composition can change dramatically. In some chronic diseases, including heart failure and chronic obstructive pulmonary disease, skeletal muscles undergo a shift toward more glycolytic, less oxidative fiber types. The mechanisms underlying this transition are complex and involve changes in muscle activity patterns, alterations in hormonal and inflammatory signaling, and possibly direct effects of hypoxia on muscle metabolism. The existence of multiple fiber types within a single muscle provides versatility. For light tasks, slow twitch motor units are recruited, providing economical force production. As tasks become more demanding, fast twitch motor units are recruited, providing additional force and speed. This allows the muscle to adapt its properties to the task at hand, using the most appropriate fibers for each situation. This specialization represents an elegant solution to competing demands. A muscle needs to be capable of both sustained, low-force contractions for posture and endurance activities, and rapid, high-force contractions for power activities. No single fiber type can optimally serve both demands. By including multiple fiber types, muscles can be jacks of all trades, if not masters of any one. And by recruiting these fiber types in an orderly fashion, from most to least economical, the nervous system optimizes performance and efficiency across a wide range of tasks. But muscle fibers, no matter how specialized or how well controlled, cannot produce useful movement unless they're attached to something. A muscle fiber contracting in isolation generates force but produces no motion. For muscles to move the body, they must attach to bones through tendons, and the geometry of these attachments profoundly influences how muscles function. So we turn now to the subject of muscle attachments, origins, insertions, and the connective tissues that transmit force from muscle to skeleton. Muscle, for all its contractile sophistication, would be functionally useless without secure attachments to the skeleton. The force generated within sarcomeres must be transmitted through the muscle fiber, through connective tissue, across tendons, and finally to bone. Only then can muscle contraction produce movement at joints. The nature of these attachments, their location, and their mechanical properties profoundly influence muscle function. Most skeletal muscles attach to bones at 2 or more points. By convention, one attachment is called the origin and the other the insertion, though these terms are somewhat arbitrary and their precise definitions vary depending on who you ask. Traditionally, The origin is defined as the more proximal or more fixed attachment point, while the insertion is the more distal or more mobile attachment. The muscle is said to originate from one bone and insert onto another. For example, the biceps brachii is described as originating from the scapula, the shoulder blade, and inserting on the radius, one of the bones of the forearm. When the biceps contracts, the insertion on the radius moves substantially, pulling the forearm toward the shoulder, while the origin on the scapula moves little, if at all. This naming convention works well for many limb muscles where there is a clear distinction between fixed and mobile attachments. However, the convention breaks down in some situations. Consider a muscle that spans between 2 points on the trunk, where neither attachment is obviously more fixed than the other. Or consider that the same muscle might pull from different directions depending on body position. During a pull-up, the biceps pulls the body upward toward the fixed hands, effectively reversing the usual relationship between origin and insertion. For these reasons, some anatomists prefer more neutral terminology, referring simply to attachments, proximal and distal attachments, or medial and lateral attachments, avoiding the potentially misleading implications of origin and insertion. Nevertheless, the traditional terminology persists and remains useful for many muscles where the distinction is clear and functionally meaningful. The physical connection between muscle and bone occurs through tendons or, in some cases, through aponeuroses. Tendons are cord-like or strap-like structures composed primarily of collagen, a protein that provides enormous tensile strength. The collagen is organized into parallel bundles of fibers, aligned along the direction of force transmission, creating a structure that is strong in tension but flexible, allowing the tendon to bend around bony prominences or pass through pulleys without breaking. At one end, the tendon connects to muscle at the myotendinous junction. This is a highly specialized interface where the muscle fibers interdigitate with the tendon, creating a large surface area for force transmission. The sarcolemma of the terminal muscle fibers forms deep folds that project into the tendon, while collagen fibers from the tendon penetrate between the muscle fibers. This complex three-dimensional structure distributes forces across a large area, preventing the muscle fibers from tearing away from the tendon during contraction. At the molecular level, the connection between muscle and tendon involves several key proteins. Inside the muscle fiber, the contractile apparatus is connected to the sarcolemma through protein complexes that include dystrophin, integrins, and various associated proteins. These proteins span the sarcolemma, connecting the actin filaments on the inside to the extracellular matrix on the outside. The extracellular matrix of the endomysium, which surrounds each muscle fiber, is continuous with the collagen of the tendon, creating a continuous path for force transmission from sarcomere to bone. At the other end of the tendon, the connection to bone occurs through another specialized interface. The tendon fibers don't simply attach to the surface of the bone. Instead, they penetrate into the bone, with collagen fibers extending into the bone matrix, creating an extremely strong attachment. At the bone-tendon interface, there's a transition zone where the tendon gradually mineralizes, transitioning from unmineralized collagen to mineralized fibrocartilage to bone. This gradual transition helps distribute forces and prevents stress concentrations that could cause the tendon to tear away from the bone. Tendons vary considerably in size and shape, depending on the muscle and its function. Some tendons are quite short, only a few millimeters long, while others are remarkably long. The plantaris muscle, a small muscle in the calf, has a tendon that extends over 30 centimeters, far longer than the muscle belly itself. The tendons of the finger flexors extend from the forearm through the palm and into the fingers, spanning 15 centimeters or more. Some muscles have very broad, flat attachments rather than distinct tendons. These sheet-like connective tissue structures are called aponeuroses. The external oblique muscle of the abdomen, for instance, has a broad aponeurosis that forms part of the anterior abdominal wall. The advantage of an aponeurosis is that it distributes forces over a large area, reducing stress on any particular point. The disadvantage is that it doesn't allow the force to be focused on a specific point or transmitted over a long distance as a tendon does. The mechanical properties of tendons are critical to muscle function. Tendons are strong, with tensile strengths of 50 to 100 megapascals or more, comparable to nylon rope. This strength is essential given the forces muscles can generate. A large muscle, like the quadriceps, can produce forces exceeding 3,000 newtons, roughly 700 pounds of force. The patellar tendon, which transmits this force from the quadriceps to the tibia, must be able to withstand these enormous loads repeatedly without failing. But tendons aren't just strong, they're also elastic. When loaded, a tendon stretches, storing elastic energy like a spring. This elasticity serves several important functions. First, it helps protect muscles from damage during eccentric contractions, when the muscle is forcibly lengthened while contracting. The tendon absorbs some of the mechanical energy, reducing the stress on the muscle fibers themselves. Second, the elastic energy stored in tendons during one phase of a movement can be released to enhance force production during the next phase, improving efficiency. This elastic energy storage and return is particularly important during activities like running or jumping. When you land from a jump, the impact force stretches the tendons of your leg muscles, storing elastic energy. When you push off again, this stored energy is released, contributing to the force production without requiring additional metabolic energy. Kangaroos have taken this principle to an extreme, with extremely long, elastic tendons in their legs that allow them to hop efficiently at high speeds, storing and releasing elastic energy with each hop. The amount of stretch that occurs in a tendon depends on its stiffness, which varies considerably among tendons. Stiffer tendons stretch less for a given force, which is advantageous when rapid, precise control is needed. The tendons controlling finger movements are quite stiff, allowing precise control of finger position. More compliant tendons stretch more, which is advantageous for elastic energy storage. The Achilles tendon, which connects the calf muscles to the heel, is relatively compliant and stores substantial elastic energy during running and jumping. Tendon stiffness can adapt to training. Regular loading, as occurs during strength or plyometric training, increases tendon stiffness by increasing collagen content and altering the organization of collagen fibers. This adaptation improves force transmission but reduces energy storage capacity, a trade-off that reflects the specific demands being placed on the system. The moment arm of a muscle, the perpendicular distance from the muscle's line of action to the joint's axis of rotation, determines the torque the muscle can produce for a given force. A larger moment arm means more torque for the same muscle force. Moment arm also changes as the joint moves. For most muscles, the moment arm is smallest at the extremes of joint motion and largest in the mid-range. This creates a mechanical disadvantage at extreme joint angles that compounds the disadvantage from the length-tension relationship of the muscle fibers. Some muscles have insertions that create particularly favorable or unfavorable mechanical leverage. A muscle with a more distal insertion has a longer moment arm and produces more torque for a given force, which is mechanically advantageous for lifting loads. However, it must shorten more to produce a given amount of joint movement, which is mechanically disadvantageous for producing speed. Different species and different individuals within a species show variation in muscle attachment points, likely reflecting evolutionary trade offs between force and speed for different ecological niches or activity patterns. Tendons and aponeuroses can be injured, particularly during activities that involve high forces or rapid stretching. Tendinitis, inflammation of a tendon, occurs when the tendon is loaded beyond its capacity to repair the microscopic damage that occurs during normal use. The inflammation causes pain and can impair function. Chronic overloading can lead to tendinosis, a degenerative condition where the normal collagen structure of the tendon breaks down and is replaced with disorganized, weaker tissue. Tendons can rupture completely, particularly if suddenly overloaded. Achilles tendon rupture is relatively common in middle-aged individuals who engage in sudden, intense physical activity without adequate conditioning. The rupture is often described as feeling like being kicked in the back of the leg, followed by immediate inability to plantar flex the foot or rise up on the toes. Surgical repair is often necessary, though outcomes are generally good with appropriate treatment. The myotendinous junction is also vulnerable to injury. Muscle strains, commonly called pulled muscles, often occur at this interface rather than within the muscle belly itself. The sudden change in mechanical properties from compliant muscle to stiff tendon creates a stress concentration, and rapid or excessive loading can cause tearing at this site. Grade 2 strains involve microscopic tearing with minimal loss of function. Grade 2 strains involve partial tearing with noticeable loss of strength and range of motion. Grade 3 strains are complete ruptures of the muscle tendon unit, requiring surgical repair. Understanding muscle attachments is essential for understanding not just how muscles produce movement, but also how they can be injured and how injuries can be treated. The connective tissue structures that link muscle to bone are not passive cables but adaptive, living tissues that respond to the mechanical demands placed on them. They're essential components of the neuromuscular system, deserving consideration alongside the muscle fibers themselves. But muscles do not work in isolation, pulling in random directions. They work in coordinated groups, with some muscles producing the primary movement, others assisting, others opposing, and still others stabilizing joints to allow efficient force production. This coordination, the symphony of agonists, antagonists, synergists, and stabilizers, is the subject we must now explore to understand how individual muscle contractions are orchestrated into functional movement. Synergists are muscles that assist the agonist, either by contributing additional force in the same direction or by neutralizing unwanted actions of the agonist. Many muscles don't produce pure, single-axis movements. When they contract, they may produce a desired primary action along with secondary, undesired actions. Synergists can contract to cancel out these secondary actions, allowing the agonist's primary action to occur in isolation. Take the biceps brachii as an example again. Its primary action is elbow flexion, but it also supinates the forearm, rotating it so the palm faces upward. If you want to flex your elbow while keeping your palm facing downward in pronation, the pronator muscles must act as synergists, contracting to neutralize the biceps supination effect while allowing its flexion action to proceed. Without these synergists, you couldn't flex your elbow while maintaining forearm pronation. The supination would occur whether you wanted it or not. Fixators are a specialized type of synergist that stabilize one bone so that another muscle can pull on a different bone. When you do a push-up, your arm muscles push your body away from the ground. But for this to work, your scapula must be held stable against your rib cage. Multi-joint movements add another layer of complexity. When multiple joints move simultaneously, the forces generated at one joint affect the other joints. This is called dynamic coupling or interaction torque. If you flex your elbow rapidly, the angular acceleration of the forearm creates reaction forces that tend to extend the shoulder. To produce a pure elbow flexion without shoulder extension, the shoulder muscles must activate to counteract these interaction forces. The nervous system becomes quite skilled at anticipating and compensating for these interaction forces. When you perform a well-learned multi-joint movement, the activation patterns of muscles at different joints are precisely coordinated, with muscles activating not just to move their own joint, but to compensate for forces arising from movement at other joints. This coordination allows for smooth, efficient movement. Reciprocal inhibition is a neural mechanism that facilitates coordinated action of agonists and antagonists. When a motor neuron activating an agonist is stimulated, inhibitory interneurons in the spinal cord simultaneously inhibit the motor neurons of the antagonist muscles. This automatic inhibition helps the antagonist relax when the agonist contracts, making movement smoother and more efficient. Reciprocal inhibition can be overridden by descending commands from the brain when co-contraction is desired, but it provides a default pattern that simplifies motor control. The stretch reflex also plays a role in coordinated movement. When a muscle is stretched rapidly, sensory receptors called muscle spindles detect the stretch and trigger a reflex contraction of the same muscle. This monosynaptic reflex, the simplest reflex in the body, helps resist unexpected stretches and contributes to muscle tone. Simultaneously, reciprocal inhibition ensures that the antagonist is inhibited, allowing the stretched muscle to contract without opposition. Muscle spindles are fascinating sensory structures embedded within the muscle belly, running parallel to the main muscle fibers. Each spindle contains specialized muscle fibers called intrafusal fibers, as opposed to the standard extrafusal fibers that generate force. The intrafusal fibers have sensory nerve endings wrapped around their central region, detecting stretch. They also have motor innervation at their ends, allowing the nervous system to adjust the sensitivity of the spindle. When the muscle is stretched, the spindle is also stretched, and the sensory endings fire, sending signals to the spinal cord. This triggers the stretch reflex, causing the muscle to contract and resist the stretch. But here's the clever part. When the muscle contracts voluntarily, the intrafusal fibers are also activated by their motor neurons, called gamma motor neurons. This maintains tension in the spindle even as the main muscle shortens, keeping the spindle sensitive throughout the range of motion. Without this gamma motor neuron activation, the spindle would go slack whenever the muscle contracted voluntarily, making it insensitive to stretch. By activating alpha motor neurons to the main muscle fibers and gamma motor neurons to the spindle fibers simultaneously, the nervous system ensures that the spindle remains functional as a length sensor, regardless of whether the muscle is contracting or relaxed. This is called alpha-gamma coactivation, and it's a fundamental feature of motor control. Golgi tendon organs are another sensory structure, located in the tendons where muscle attaches to bone. Unlike muscle spindles, which sense muscle length, Golgi tendon organs sense muscle tension. When a muscle generates force, it pulls on the tendon, and the Golgi tendon organ detects this pulling force. The sensory signals from Golgi tendon organs go to the spinal cord, where they inhibit the muscle's own motor neurons and facilitate antagonist motor neurons. This negative feedback loop helps prevent excessive muscle force that might damage the muscle or tendon. If the muscle generates too much force, the Golgi tendon organ signals become strong enough to inhibit the muscle, reducing force. This is sometimes called the inverse stretch reflex or the tendon reflex, and it's protective in nature. However, this reflex can be overridden by voluntary commands, allowing us to generate very high forces when necessary, though at some risk of injury. The coordination of multiple muscles extends beyond individual joints to whole body movements. During walking, for instance, muscles throughout both legs, the trunk, and the arms must activate in precise temporal patterns. The sequence of muscle activations is so complex that it's been the subject of hundreds of research studies, yet we perform it automatically, without thinking. This automaticity reflects the existence of central pattern generators, neural circuits in the spinal cord that can produce rhythmic, coordinated patterns of muscle activation without continuous input from the brain. Central pattern generators were first discovered in animals like cats and lampreys, where researchers found that even after disconnecting the spinal cord from the brain, the spinal cord could still produce coordinated locomotor patterns. In humans, central pattern generators are less independent, requiring supraspinal input to initiate and modulate their activity, but they still handle much of the detailed coordination of rhythmic movements like walking, freeing the brain to focus on navigation, balance, and adjusting the gait to terrain and circumstances. The concept of muscle synergies takes this idea of coordination even further. Rather than controlling each muscle independently, the nervous system appears to control muscles in groups, with a single neural command activating a synergy, a characteristic pattern of activation across multiple muscles. Evidence for this comes from studies using dimensionality reduction techniques, which find that the complex patterns of muscle activation during movement can be explained by a relatively small number of underlying synergies, typically 5 to 10 for complex tasks. These synergies are thought to simplify motor control, reducing the number of independent signals the brain must generate. Rather than specifying the activation of hundreds of muscles individually, the brain can select and combine a smaller number of synergies, much like combining primary colors to create a wide range of hues. This reduces the computational burden and may make motor learning easier, since learning can involve discovering new combinations of existing synergies rather than learning entirely new patterns from scratch. Of course, whether synergies are truly a control strategy used by the nervous system or simply a mathematical description of correlated muscle activity is still debated. But the observation that muscle activation patterns are lower dimensional than the number of muscles suggests that there is indeed some underlying organization, some structure in how the nervous system coordinates the muscular system. In pathological conditions, coordination can break down. After a stroke, for instance, patients often exhibit abnormal co-contraction patterns, with agonists and antagonists contracting together more than they should. This makes movement stiff, slow, and effortful. Spasticity, increased muscle tone, and exaggerated reflexes is common after upper motor neuron lesions, reflecting the loss of descending inhibitory control over spinal reflexes. Cerebellar damage impairs the coordination of multi-joint movements, leading to dysmetria, movements that overshoot or undershoot their targets, and dysdiadochokinesia, difficulty performing rapid alternating movements. Understanding coordinated muscle action is essential for understanding motor control, motor learning, and motor rehabilitation. Individual muscles are the building blocks, but movement arises from their coordinated action. The nervous system doesn't think in terms of individual muscles. It thinks in terms of movements, goals, and synergies. The muscles are simply the instruments through which these goals are achieved. But muscles, coordinated or not, don't all contract in the same way. Depending on the task, a muscle might shorten while generating force, maintain constant length while generating force, or even lengthen while generating force. These different types of contractions, isotonic, isometric, and eccentric, have different mechanical properties and different functional roles. Understanding these different contraction types and the relationships between force, velocity, and length is essential to understanding muscle function in real-world tasks, which we'll explore in the next chapter. The term contraction, as applied to muscle, is somewhat misleading. In everyday language, to contract means to become smaller, to shorten, and indeed, muscles often do shorten when they contract, pulling their attachments closer together. But muscles can also generate force without shortening, or even while being forcibly lengthened. The term contraction, in the physiological sense, refers to the active generation of force through cross-bridge cycling, regardless of whether the muscle actually shortens, stays the same length, or lengthens. This leads to three broad categories of muscle contraction, classified by whether the muscle changes length during force production. Isometric contractions occur when the muscle generates force but doesn't change length. Isotonic contractions occur when the muscle changes length while generating relatively constant force. Isotonic contractions are further divided into concentric contractions, where the muscle shortens, and eccentric contractions, where the muscle lengthens. Isometric contractions, from the Greek iso meaning same and metric meaning length, occur when you push or pull against an immovable object, or when you hold a weight steady in a fixed position. The muscle is activated, cross bridges are cycling, ATP is being consumed, and force is being generated, but the muscle length remains constant because the external force exactly balances the muscle force. No movement occurs, and no external work is performed, at least not in the physics sense of work as force times distance. Despite the lack of external work, isometric contractions are metabolically expensive and can be quite fatiguing. The sustained activation requires continuous ATP consumption to maintain cross-bridge cycling and to pump calcium back into the sarcoplasmic reticulum. Blood flow to the muscle may be partially or completely occluded when intramuscular pressure exceeds arterial pressure, limiting oxygen delivery and waste removal. This is why holding a weight steady in a fixed position or maintaining a static posture like a plank exercise becomes increasingly uncomfortable and eventually impossible as fatigue develops. The force a muscle can generate isometrically depends on muscle length, reflecting the length-tension relationship we discussed earlier. At optimal length, where overlap between thick and thin filaments is maximal, isometric force is greatest. At shorter or longer lengths, force decreases. For most muscles in the body, the resting length, the length at which the muscle sits when the body is in an anatomically neutral position, is close to optimal length. This ensures that muscles can generate near maximal forces during typical movements. Isometric training, where exercises are performed against immovable resistance or by holding weights in fixed positions, can increase strength, though the strength gains tend to be specific to the joint angle at which training occurred. If you train isometric strength at 90 degrees of elbow flexion, you'll gain strength at that angle, but you may not gain as much strength at other angles. This angle specificity is less pronounced with isotonic training, which involves movements through a range of motion and trains the muscle across multiple lengths. Concentric contractions occur when a muscle shortens while generating force. This is what most people think of as muscle contraction. When you lift a weight, curl a dumbbell, or stand up from a chair, the involved muscles are contracting concentrically. The muscle force is greater than the external resistance, so the muscle shortens, and movement occurs in the direction the muscle is pulling. During concentric contractions, the muscle performs positive work, using metabolic energy to move an external load. The relationship between force and velocity during concentric contractions is inversely related. As the velocity of shortening increases, the force the muscle can generate decreases. This force-velocity relationship is one of the fundamental properties of muscle. At zero velocity, during an isometric contraction, the muscle can generate maximum force. As shortening velocity increases, force declines, following a hyperbolic curve. At very high velocities, approaching the maximum shortening velocity of the muscle, force drops to nearly zero. This explains why it's easier to lift a weight slowly than quickly, and why rapid movements require lighter loads than slow movements. The force-velocity relationship exists because of the kinetics of cross-bridge cycling. During rapid shortening, the myosin heads have less time in contact with actin during each cycle. They attach, perform a power stroke, and detach quickly, limiting the force each cycle can generate. Additionally, some myosin heads may be forcibly detached before completing their power stroke, further reducing force. At slower shortening velocities, more cross bridges are attached at any moment, and each completes a fuller power stroke, generating more force. Power, defined as force times velocity, is maximized at intermediate velocities, typically around 30% of maximum velocity. This optimal power velocity represents the best compromise between force, which is high at low velocities, and velocity itself, which is high at low forces. Training for power, as required in sports involving jumping, throwing, or sprinting, often targets this optimal velocity range, using moderate loads that can be moved rapidly. Eccentric contractions occur when a muscle lengthens while generating force, essentially when the muscle is being overpowered by an external force but is resisting that force. This happens when you lower a weight, walk downhill, or land from a jump. The external force, whether gravity pulling on a weight, the ground pushing on your legs during landing, or your body weight descending a hill, is greater than the muscle force. So the muscle is forcibly lengthened even as it contracts. Eccentric contractions are metabolically efficient, requiring less ATP per unit of force produced than concentric or isometric contractions. During eccentric contraction, the external force is performing negative work on the muscle, essentially forcing the cross bridges to detach and re-extend. This mechanical energy input reduces the metabolic cost of generating force. Interestingly, this means that lowering a weight requires less metabolic energy than lifting the same weight, even though the muscular effort may feel similar. The force a muscle can generate eccentrically is greater than the force it can generate isometrically or concentrically, typically about 20 to 50% greater. This is partly because fewer cross bridges need to be active to produce a given force when the muscle is being forcibly stretched, and partly because of passive resistance from elastic elements within the muscle, including titin and the connective tissue. This greater force capacity during eccentric contraction has practical implications. You can lower more weight than you can lift, and you can walk down a steeper hill than you can walk up. However, eccentric contractions are also more damaging to muscle tissue than concentric contractions. The forcible stretching of active cross bridges and sarcomeres can cause microscopic tears in muscle fibers, disruption of the cytoskeleton, and damage to the sarcolemma. This damage triggers an inflammatory response and leads to delayed onset muscle soreness, the muscle pain and stiffness that typically peaks 24 to 48 hours after unaccustomed eccentric exercise. While this damage might seem purely negative, it's actually a stimulus for adaptation. The muscle responds to eccentric exercise-induced damage by increasing protein synthesis, adding more contractile proteins and structural proteins, leading to muscle hypertrophy and increased strength. The damage also prompts adaptations that make the muscle more resistant to future damage, a phenomenon called the repeated bout effect. After one bout of eccentric exercise, subsequent bouts cause less damage and soreness, even weeks or months later. Plyometric training exploits the unique properties of eccentric contractions. Plyometric exercises involve a rapid eccentric contraction immediately followed by a rapid concentric contraction, such as in a depth jump, where you step off a box, land, and immediately jump upward. During the landing phase, the muscles contract eccentrically, stretching tendons and storing elastic energy. During the takeoff phase, this elastic energy is released, supplementing the force from concentric contraction and enhancing performance. The effectiveness of plyometric exercises depends on minimizing the delay between eccentric and concentric phases. If the delay is too long, the stored elastic energy dissipates as heat, and the benefit is lost. The optimal coupling time is typically less than 0.2 seconds. This requires not just strong muscles, but also stiff tendons that can store and release elastic energy rapidly, and a well-trained nervous system that can coordinate the rapid transition from eccentric to concentric contraction. The distinction between these contraction types has practical implications for training and rehabilitation. Eccentric training is particularly effective for hypertrophy and strength development, but must be progressed carefully to avoid excessive soreness and injury risk. Isometric training can be valuable for developing strength at specific joint angles or for training around injuries where range of motion is limited. Concentric training is the most intuitive and forms the basis of most traditional strength training programs. The force length and force velocity relationships impose fundamental constraints on muscle function. A muscle cannot generate maximum force at very short or very long lengths, and it cannot generate maximum force at very high velocities. These constraints shape how movements are performed and how training should be structured. Understanding these relationships helps explain why movements feel easier or harder at different joint angles, why explosive movements require lighter loads than slow movements, and why some movements cause more muscle soreness than others. Yet, these individual muscles and their various contraction types don't operate in a vacuum. They're attached to bones through tendons, and the geometry of these attachments, the angles at which forces are applied, and the distances over which forces act determine the mechanical advantage or disadvantage of muscle action. The body operates as a system of levers, with bones as the rigid bars, joints as the fulcrums, and muscles providing the effort forces. Understanding these lever systems is essential to understanding how muscles actually move the skeleton. When muscles contract, they pull on bones. This much is straightforward. What's less obvious is that this pulling action operates according to the same principles that govern any lever system, the kind you might have encountered in a high school physics class and promptly forgotten. The human body, it turns out, is a collection of levers, each one optimized for particular tasks. Some are designed to maximize force, others to maximize speed or range of motion. Understanding these mechanical arrangements helps explain why certain movements come easily while others require considerable effort, and why the architecture of the musculoskeletal system is arranged the way it is. A lever is essentially a rigid bar that moves around a fixed point called a fulcrum. In the body, bones serve as the rigid bars, joints act as fulcrums, and muscles provide the effort needed to move the system. There's also a load or resistance to overcome, which might be the weight of the limb itself, an object being lifted, or any external force acting on the body. These three components, effort, fulcrum, and load, can be arranged in different configurations, giving rise to three classes of levers. Each class has distinct mechanical properties and appears in different locations throughout the body. First-class levers place the fulcrum between the effort and the load. Picture a seesaw with the pivot point in the middle. One side goes down while the other goes up. In the human body, a classic example is the head resting on the vertebral column, the atlanto-occipital joint, where the skull meets the first cervical vertebra, serves as the fulcrum. The weight of the face and anterior skull acts as the load, pulling the head forward and downward. Meanwhile, the posterior neck muscles provide the effort, pulling back to keep the head upright. This arrangement is why maintaining good posture requires continuous muscular effort. Without it, your head would simply tip forward under its own weight, an all too familiar sensation during long, dull meetings. Another example of a first-class lever is the action of the triceps brachii during elbow extension when the hand is pushing against resistance. The elbow joint is the fulcrum. The triceps provides effort at its insertion on the olecranon process of the ulna, and the resistance is located at the hand. First-class levers can favor either force or speed, depending on where the fulcrum sits relative to the effort and load. If the fulcrum is closer to the load, the system favors force. If it's closer to the effort, the system favors speed and range of motion. The body employs both strategies depending on the task at hand. Second-class levers position the load between the fulcrum and the effort. A wheelbarrow is the classic non-biological example. The wheel acts as the fulcrum, the load sits in the basin, and you provide effort by lifting the handles. This arrangement always favors force over speed because the effort arm, the distance from the fulcrum to where the effort is applied, is longer than the resistance arm. The distance from the fulcrum constitutes the load. In the human body, second-class levers are relatively rare, but they do exist. The most commonly cited example is standing on your toes, a movement called plantar flexion. The fulcrum is the ball of the foot, specifically the metatarsophalangeal joints, where the toes meet the rest of the foot. The load is the body weight, acting downward through the tibia onto the ankle joint. The effort comes from the calf muscles, primarily the gastrocnemius and soleus, which pull upward on the calcaneus, the heel bone, via the Achilles tendon. Because the effort is applied behind the load, This creates a second-class lever. The mechanical advantage here allows the calf muscles to lift the entire body weight with relative efficiency. This is why you can rise onto your toes repeatedly without immediate fatigue, assuming you're in reasonable shape. The leverage works in your favor. Some biomechanics texts debate whether this example truly qualifies as a second-class lever or whether it shifts into a different classification depending on the phase of movement. These are the sorts of arguments that occupy the more pedantic corners of kinesiology conferences. For practical purposes, it's sufficient to recognize that the arrangement provides a mechanical advantage, making it easier to lift your body weight than it would be without the lever system in place. Third-class levers are by far the most common arrangement in the human body, which is interesting because they operate at a mechanical disadvantage. In a third-class lever, the effort is applied between the fulcrum and the load. Picture a pair of tweezers or chopsticks. You apply force in the middle, the pivot is at one end and the load is at the other. This setup requires more effort to move a given load, but it offers a significant trade-off: speed and range of motion. Consider the biceps brachii during elbow flexion. The elbow joint serves as the fulcrum. The biceps attaches to the radius at the radial tuberosity, a point very close to the elbow. The load, whether it's the weight of your forearm or a dumbbell in your hand, is located much farther from the fulcrum. Because the effort is applied closer to the fulcrum than the load is, you need to generate considerably more force in the muscle than the actual weight being lifted. If you're curling a 10-kilogram weight, the biceps may need to produce 70 or 80 kilograms of force, depending on the angles involved. This seems inefficient, and in terms of pure force production, it is. But the advantage is clear when you consider movement. A small contraction of the biceps produces a much larger displacement at the hand. This amplification of movement allows for quick, sweeping motions. You can reach out and grab something rapidly, throw a ball with speed, or perform intricate hand movements, all thanks to third-class lever systems. The muscular system sacrifices mechanical advantage for versatility and velocity, a trade-off that serves us well in a world where quick reactions and dexterous manipulation are often more valuable than brute strength alone. Most limb movements involve third-class levers. The brachialis, another elbow flexor, operates on the same principle. So does the brachioradialis. When you flex your hip, the iliopsoas muscle pulls on the femur close to the hip joint, moving the entire leg through a wide arc. When you flex your shoulder, the anterior deltoid applies effort near the shoulder joint to swing the arm forward. In each case, the muscle works harder than it would in a more mechanically advantageous system, but the benefit is speed, range, and the ability to position the limbs precisely in space. The concept of mechanical advantage can be quantified. It's the ratio of the effort arm to the resistance arm. If the effort arm is longer than the resistance arm, the mechanical advantage is greater than 1, meaning you're amplifying force. This is the case in second-class levers. If the effort arm is shorter, the mechanical advantage is less than 1, meaning you're amplifying distance and speed at the expense of force. This describes third-class levers. First-class levers can go either way, depending on the specific arrangement. Another factor to consider is the angle of muscle insertion. Muscles don't always pull in a perfectly perpendicular direction relative to the bone they're moving. The angle of pull affects the efficiency of force transmission. When a muscle pulls at a 90-degree angle to the bone, it produces the maximum rotational force, or torque. As the angle deviates from 90 degrees, some of the muscle's force is wasted either pulling the bone into the joint or pulling it away rather than rotating it. This is why certain portions of a movement feel easier than others. During a bicep curl, the middle portion of the lift, where the forearm is roughly perpendicular to the upper arm, feels relatively strong. At the very beginning or end of the movement, where the angle is less favorable, the same weight feels heavier. The length of the lever arm also changes during movement. In biomechanics, the moment arm refers to the perpendicular distance from the line of muscle force to the joint's axis of rotation. As a joint moves through its range of motion, this distance changes, altering the muscle's leverage. This is one reason why muscles work in groups. At different points in a movement, different muscles contribute more or less to the effort depending on their current leverage. It's a finely tuned collaboration, one that generally proceeds without conscious oversight, which is probably for the best. The human body also employs strategies to improve leverage where it's needed. Sesamoid bones, small bones embedded within tendons, can increase the angle of pull and thus improve mechanical advantage. The patella, or kneecap, is the largest sesamoid bone. It sits within the quadriceps tendon and increases the distance between the tendon and the knee joint's axis of rotation. This increases the moment arm, allowing the quadriceps to produce greater torque during knee extension. Without the patella, extending the knee against resistance would require significantly more muscular effort. Other, smaller sesamoid bones appear in the hands and feet, serving similar functions on a smaller scale. Leverage also explains why longer limbs can be advantageous or disadvantageous depending on the activity. In activities that favor speed and reach, such as swimming or basketball, longer limbs provide a mechanical benefit. The longer lever arm amplifies the distance traveled by the end of the limb for a given amount of muscle contraction. They require less torque to move quickly and are easier to control through complex rotations. The concept of leverage also has implications for injury. Joints that operate at a mechanical disadvantage are under greater stress. The forces within the joint can be several times greater than the external load being moved. In the shoulder, for example, the supraspinatus muscle helps initiate arm abduction, lifting the arm away from the body. Because of the poor leverage, especially in the early degrees of abduction, the muscle must generate substantial force. This high demand contributes to the frequency of rotator cuff injuries, particularly in activities that involve repetitive overhead motion. Similarly, the lower back operates under considerable mechanical strain. When you bend forward to lift something, the erector spinae muscles must work against a long lever arm, your entire upper body cantilevered out in front of the spine. Even lifting a relatively light object can create enormous compressive forces on the lumbar vertebrae and discs. This is why proper lifting technique emphasizes bending at the knees and hips rather than the waist. By shortening the lever arm and engaging the stronger muscles of the legs, you reduce the strain on the lower back. Rehabilitation and exercise science make extensive use of leverage principles. Changing the position of a weight relative to a joint can make an exercise easier or harder without changing the actual load. For example, a plank performed with arms extended is harder than one performed on the forearms because the longer lever arm increases the torque that the core muscles must resist. A leg raise performed with straight legs is more challenging than one with bent knees for the same reason. Physical therapists manipulate these variables to progressively load injured tissues, starting with shorter lever arms and advancing to longer ones as strength and healing improve. Even something as simple as using a doorknob involves leverage. The knob is placed far from the hinge, the fulcrum, to maximize the effect of your effort. You apply a small force at the knob, and the door swings open relatively easily. If the knob were placed near the hinge, you'd need to push much harder to achieve the same result. The same principle applies throughout the body. Muscles generally insert close to joints, creating third-class levers that favor speed and range over force. When force is needed, the body relies on recruitment of additional motor units or co-contraction of synergist muscles rather than changing the lever system itself, which is, of course, fixed by anatomy. There's an elegance to the way these systems are arranged. The body doesn't optimize for a single variable. It balances competing demands: strength, speed, endurance, precision, and range of motion. The lever systems reflect these compromises. Where force is paramount, as in the calf muscles lifting the body, we find second-class levers. Where speed and reach matter more, as in the limbs, third-class levers dominate. And where balance is needed, first-class levers provide a middle ground. It's worth noting that while these mechanical principles are fixed by the skeleton structure, muscle strength and coordination can be trained. You can't change the length of your bones or the insertion points of your muscles, but you can increase the force your muscles produce and improve the timing and efficiency of their contractions. This is the essence of strength training and skill development. You're working within the constraints of your lever systems, but optimizing the force-generating capacity and control of the muscles operating those levers. In the end, the muscular system's relationship with the skeletal system is one of elegant collaboration. Muscles provide the force, bones provide the rigid structure, joints provide the pivot points, and the resulting lever systems allow for an astonishing range of movements. From the delicate manipulation of a surgical instrument to the explosive power of a sprinter leaving the blocks, these mechanical principles are at work. They operate quietly, reliably, and for the most part, without any need for conscious awareness. Which is just as well, because calculating torques and moment arms in real time would make even the simplest movements unbearably tedious. Up to this point, we've focused primarily on skeletal muscle, the voluntary system that moves limbs and maintains posture. But skeletal muscle represents only one branch of the muscular system. The other two, smooth muscle and cardiac muscle, operate largely without conscious input. They govern processes that, if left to voluntary control, would be disastrously unreliable. Imagine having to remember to make your heart beat or consciously regulate the diameter of your blood vessels. It would be a full-time occupation with little margin for error. Fortunately, these tasks are handled by involuntary muscle, systems that function autonomously, adjusting continuously to the body's needs without requiring attention or intention. Smooth muscle is found throughout the body, lining the walls of hollow organs and structures. It appears in the digestive tract, blood vessels, airways, bladder, uterus, and even in the small muscles attached to hair follicles in the skin. Despite its widespread distribution, smooth muscle tends to go unnoticed until something goes wrong. A bout of gastrointestinal cramping, a spike in blood pressure, or the onset of labor contractions. These are moments when smooth muscle makes its presence felt. The rest of the time, it works quietly in the background, adjusting tension, propelling contents, and regulating flow. Structurally, smooth muscle differs considerably from skeletal muscle. Its cells are smaller, spindle-shaped, and contain a single centrally located nucleus. There are no striations, no organized sarcomeres arranged in neat rows. Instead, actin and myosin filaments are present, but arranged in a less orderly fashion, anchored to structures called dense bodies, scattered throughout the cell and along the cell membrane. These dense bodies serve a similar function to the Z discs in skeletal muscle, providing attachment points for the contractile filaments. When myosin pulls on actin, the dense bodies are drawn together, causing the cell to shorten and generate force. The absence of a highly organized sarcomere structure gives smooth muscle some unique properties. It can contract over a much wider range of lengths compared to skeletal muscle, which is useful in organs that need to accommodate varying volumes. The stomach, for example, can stretch considerably when full and contract down when empty, all while maintaining the ability to generate force. The bladder does much the same. Smooth muscle is well suited to these tasks, able to maintain tension across a broad spectrum of lengths without losing effectiveness. Smooth muscle cells are connected to one another by gap junctions, specialized channels that allow ions and small molecules to pass directly from cell to cell. This electrical coupling means that when one cell depolarizes, the signal can spread to neighboring cells, allowing coordinated contraction across a sheet of tissue. This is particularly important in organs like the intestines, where waves of contraction, known as peristalsis, propel contents along the length of the digestive tract. The coordinated nature of these contractions depends on the ability of smooth muscle cells to communicate rapidly with one another. There are two main types of smooth muscle: single unit and multi-unit. Single unit smooth muscle, also called visceral smooth muscle, is the more common type. It's found in the walls of most hollow organs, the digestive tract, uterus, and bladder. In this arrangement, large numbers of cells function as a single unit, hence the name. They're electrically coupled via gap junctions, and when one cell is stimulated, the depolarization spreads throughout the entire sheet. This produces a synchronized contraction. Single unit smooth muscle often exhibits automaticity, meaning it can generate its own action potentials without external input. Pacemaker cells within the tissue spontaneously depolarize at regular intervals, triggering waves of contraction. This intrinsic rhythm can be modulated by neural or hormonal signals, but it doesn't depend on them. Multi-unit smooth muscle, on the other hand, is composed of cells that function more independently. Each cell is separately innervated by a nerve fiber, allowing for more precise control. This type of smooth muscle is found in locations where fine adjustments are needed: the iris of the eye, the walls of large arteries, the airways of the lungs, and the muscles attached to hair follicles. In the iris, for example, multi-unit smooth muscle allows for precise regulation of pupil size in response to light levels. In the airways, it adjusts the diameter of the bronchioles to regulate airflow. The degree of control afforded by multi-unit smooth muscle makes it well suited to these tasks. Smooth muscle contraction is triggered by a rise in intracellular calcium, much like skeletal muscle, but the mechanism is different. In smooth muscle, calcium doesn't bind to troponin because smooth muscle lacks troponin. Smooth muscle is regulated by the autonomic nervous system, the branch of the nervous system that controls involuntary functions. The sympathetic and parasympathetic divisions often have opposing effects on smooth muscle. In the digestive tract, parasympathetic stimulation generally increases smooth muscle activity, enhancing peristalsis and promoting digestion. Sympathetic stimulation, conversely, tends to inhibit digestive smooth muscle, part of the body's fight or flight response that diverts resources away from digestion. In blood vessels, sympathetic stimulation typically causes vasoconstriction, narrowing the vessels and increasing blood pressure. The balance between these two systems allows for fine-tuned control over smooth muscle activity throughout the body. Hormones also play a significant role in regulating smooth muscle. Epinephrine, released from the adrenal glands during stress, can cause relaxation of smooth muscle in the airways, facilitating increased airflow to the lungs. Oxytocin, released during childbirth, stimulates powerful contractions of the smooth muscle in the uterus. Vasopressin affects smooth muscle in blood vessel walls, contributing to blood pressure regulation. Local factors, such as oxygen and carbon dioxide levels, pH, and temperature, can also influence smooth muscle tone. This multi-layered regulation allows smooth muscle to respond to a wide range of physiological demands. Cardiac muscle, the third type of muscle tissue, shares features with both skeletal and smooth muscle, but remains distinct. Like skeletal muscle, it's striated, with organized sarcomeres that give it a banded appearance under the microscope. Like smooth muscle, it's involuntary and exhibits automaticity. Cardiac muscle is found exclusively in the heart, and its sole function is to pump blood. This is not a task that allows for SUSI. From the early weeks of embryonic development until the moment of death, cardiac muscle contracts rhythmically, without pause. Over an average human lifetime, it beats billions of times, an achievement of endurance that no skeletal muscle could match. Cardiac muscle cells, called cardiomyocytes, are shorter and thicker than skeletal muscle fibers. They typically contain a single nucleus, though some may have two. The cells are branched, connecting with neighboring cells at specialized junctions called intercalated discs. These discs contain two important types of junctions, desmosomes and gap junctions. Desmosomes provide strong mechanical attachments between cells, ensuring that the force generated by one cell is effectively transmitted to its neighbors. This is crucial because the heart must function as a unified whole, with all cells pulling together, literally, to generate the pressure needed to eject blood. Gap junctions, as in smooth muscle, allow electrical signals to pass directly from cell to cell. This electrical coupling means that when one cardiomyocyte depolarizes, the signal spreads rapidly throughout the heart muscle. This allows the atria to contract in unison, followed by the ventricles, producing the coordinated pumping action necessary for effective circulation. The heart doesn't rely on individual motor units the way skeletal muscle does. Instead, it functions as a functional syncytium, a network of cells that act as one. Cardiac muscle is autorhythmic. Specialized pacemaker cells, located primarily in the sinoatrial node in the right atrium, spontaneously generate action potentials at regular intervals. This intrinsic rhythm sets the heart rate, typically around 60 to 100 beats per minute at rest in adults. The signal spreads from the SA node through the atria, causing them to contract. It then reaches the atrioventricular node, where it's briefly delayed to allow the atria to finish emptying before the ventricles contract. From the AV node, the signal travels down the bundle of His and into the Purkinje fibers, which distribute it rapidly throughout the ventricular muscle. This coordinated sequence ensures that the heart's chambers contract in the proper order, maximizing pumping efficiency. Though the heart generates its own rhythm, it's not beyond external influence. The autonomic nervous system modulates heart rate and contractility. Sympathetic stimulation increases both, preparing the body for activity. Parasympathetic stimulation, mediated by the vagus nerve, slows the heart rate and decreases contractility, promoting rest and recovery. Hormones like epinephrine and thyroid hormone can also affect cardiac function, but even in the absence of all external input, the heart continues to beat. Experiments have shown that if a heart is removed from the body and kept supplied with oxygen and nutrients, it will continue contracting on its own. This intrinsic automaticity is one of cardiac muscle's most remarkable features. Cardiac muscle contraction follows the same sliding filament mechanism as skeletal muscle. Calcium enters the cell, binds to troponin, exposes binding sites on actin, and cross-bridge cycling ensues. However, cardiac muscle relies more heavily on extracellular calcium. When an action potential reaches a cardiomyocyte, voltage-gated calcium channels in the cell membrane open, allowing calcium to flow in from the extracellular fluid. This calcium then triggers the release of additional calcium from the sarcoplasmic reticulum, a process called calcium-induced calcium release. The resulting surge in intracellular calcium initiates contraction. The duration of a cardiac muscle action potential is much longer than that of skeletal muscle, lasting 200 to 300 milliseconds compared to just a few milliseconds in skeletal muscle. This prolonged depolarization is due to a plateau phase where calcium channels remain open. The extended action potential has an important consequence. It prevents tetanus. In skeletal muscle, rapid repeated stimulation can cause sustained contraction or tetanus because the muscle doesn't have time to relax between stimuli. In cardiac muscle, the long refractory period ensures that the muscle has time to relax before it can be stimulated again. This is essential for the heart's function. The heart must relax to refill with blood between contractions. If cardiac muscle could undergo tetanus, the heart would seize up, unable to pump, and that would be the end of things rather quickly. Cardiac muscle is highly resistant to fatigue, far more so than skeletal muscle. This resistance is due to an abundance of mitochondria, which occupy about 25 to 35% of the cell volume, compared to just 2% in skeletal muscle. These mitochondria produce ATP almost entirely through aerobic respiration, using oxygen delivered by a rich capillary network. The heart has its own dedicated blood supply via the coronary arteries, and it extracts a higher percentage of oxygen from the blood than most other tissues. Even at rest, the heart extracts about 70 to 80% of the oxygen in the blood passing through it, compared to around 25% for skeletal muscle. This high oxygen extraction and reliance on aerobic metabolism make the heart extremely efficient, but also vulnerable to disruptions in blood supply. When a coronary artery becomes blocked, the affected region of heart muscle is deprived of oxygen, leading to a heart attack or myocardial infarction. The cells begin to die within minutes, underscoring the heart's dependence on continuous oxygen delivery. Both smooth and cardiac muscle illustrate the body's ability to automate essential functions. Voluntary control is useful for interacting with the environment, but it's unreliable for maintaining internal homeostasis. You can choose to hold your breath, but eventually, involuntary systems override that choice and force you to breathe. You can't will your heart to stop or your intestines to halt their rhythmic contractions, at least not under normal circumstances. This division of labor, voluntary muscle for interaction with the external world, involuntary muscle for maintaining the internal environment, is one of the body's fundamental organizational principles. The three muscle types, skeletal, smooth, and cardiac, are variations on a common theme. All rely on actin and myosin, All are triggered by calcium, and all convert chemical energy into mechanical work, but each is specialized for its particular role. Skeletal muscle is fast and powerful, but fatigues quickly. Smooth muscle is slow but tireless, able to maintain tension for extended periods. Cardiac muscle is rhythmic and enduring, designed for a lifetime of continuous contraction. Together, they enable the full range of muscular function, from the deliberate lifting of a hand to the unconscious regulation of blood flow, from the explosive power of a jump to the steady, relentless beating of the heart. It's a system built on variation, specialization, and above all, reliability, which is fortunate because the alternative would be rather inconvenient. The muscular system, for all its sophistication and reliability, is not immune to dysfunction. Muscles can waste away from disuse, tear under excessive strain, or fall victim to genetic disorders that progressively rob them of function. They can be attacked by the immune system, deprived of nerve signals, or simply decline with age. Understanding what happens when muscles fail, or adapt, or change over time provides a useful perspective on how the system normally operates. It also serves as a reminder that maintaining muscle health requires attention, particularly as the years accumulate. Muscle atrophy refers to the decrease in muscle mass and strength. It can result from disuse, denervation, or systemic illness. Disuse atrophy occurs when muscles are not regularly contracted against resistance. This might happen during prolonged bed rest, immobilization due to a cast, or even extended periods in microgravity as astronauts experience in space. Without the mechanical stimulus of regular loading, muscle fibers shrink. The body, ever efficient, reallocates resources away from tissue that isn't being used. Protein synthesis decreases, protein degradation increases, and the muscle gradually diminishes in size. The rate of atrophy can be surprisingly rapid. Studies of immobilized limbs show measurable muscle loss within just a few days. Over weeks, the loss becomes substantial. Strength declines even faster than size, suggesting that neural factors, the ability to recruit and coordinate motor units, are affected as well. This is why rehabilitation after an injury involves not just rebuilding muscle mass, but also retraining the nervous system to activate muscles effectively. The good news is that disuse atrophy is largely reversible. With appropriate resistance training, muscles can regain much of their lost size and strength, though the process takes time and effort. Denervation atrophy is more serious. It occurs when the nerve supplying a muscle is damaged or severed. Without neural input, the muscle receives no signals to contract. Even if the muscle is mechanically intact, it cannot function. Over time, denervated muscle fibers atrophy even more severely than those simply immobilized. The fibers become progressively smaller, and eventually, they may be replaced by connective tissue. If the nerve damage is permanent, the muscle may never recover. However, if the nerve can regenerate, or if the muscle can be reinnervated by sprouting axons from nearby intact nerves, some function may return, particularly if the intervention occurs relatively soon after the injury. Denervation doesn't just cause atrophy, it also leads to fibrillations, spontaneous contractions of individual muscle fibers that occur randomly and asynchronously. These are not visible on the surface of the skin, but can be detected with electromyography, a diagnostic tool that measures electrical activity in muscles. Fibrillations are a sign of denervation and help clinicians determine whether muscle weakness is due to nerve damage or to a problem with the muscle itself. Hypertrophy is the opposite of atrophy. an increase in muscle size. It occurs in response to repeated mechanical loading, such as resistance training. When muscles are subjected to high levels of tension, particularly eccentric contractions, where the muscle lengthens under load, they experience microscopic damage. This triggers a repair process that not only fixes the damage, but also adds new contractile proteins, making the muscle fibers thicker and stronger. The process is slow, requiring weeks to months of consistent training to produce noticeable changes. Hypertrophy primarily involves an increase in the size of existing muscle fibers, not an increase in the number of fibers. Humans are born with a set number of muscle fibers, and that number doesn't change significantly throughout life. There's some debate about whether hyperplasia, the creation of new muscle fibers, can occur in humans under certain conditions, but the evidence is limited and controversial. For practical purposes, muscle growth is about making existing fibers larger. The number and size of myofibrils within each fiber increase. The sarcoplasmic reticulum expands, and the muscle's capacity to generate force improves. Different types of training produce different adaptations. Low resistance, high repetition activities, like long-distance running, lead to increases in mitochondrial density, capillary networks, and oxidative enzyme activity. These adaptations improve endurance, but don't significantly increase muscle size. High resistance, low repetition activities, like weightlifting, promote hypertrophy and increase maximal force production. The body adapts specifically to the demands placed on it, a principle known as specificity of training. If you want to get better at lifting heavy things, you need to practice lifting heavy things. If you want to run farther, you need to run farther. The muscular system responds accordingly. Muscle strains and tears are common injuries, particularly in athletes and those engaged in vigorous physical activity. A strain occurs when muscle fibers are overstretched or subjected to excessive force, leading to partial tearing of the tissue. Strains are graded by severity. Grade 1 involves minor damage with little loss of strength. Grade 2 involves more extensive tearing and noticeable weakness. Grade 3 is a complete rupture of the muscle or tendon, often requiring surgical repair. Compartment syndrome is a painful and potentially serious condition that occurs when pressure builds up within a muscle compartment. Muscles are organized into compartments, groups of muscles surrounded by thick connective tissue sheaths called fascia. These compartments don't expand easily. If swelling occurs within a compartment, perhaps due to injury, overuse, or bleeding, the pressure can rise to dangerous levels. This increased pressure compresses blood vessels, reducing blood flow to the muscles and nerves within the compartment. If not relieved, the tissue can become ischemic, starved of oxygen, and permanent damage can result. Acute compartment syndrome is a medical emergency, often resulting from trauma such as fractures or crush injuries. The affected limb becomes swollen, tense, and extremely painful, particularly when the muscles are passively stretched. The pain is often out of proportion to the apparent injury, a key warning sign. Treatment involves surgically opening the fascia to relieve the pressure, a procedure called a fasciotomy. If performed promptly, full recovery is possible. If delayed, the muscles and nerves can suffer irreversible damage, leading to permanent weakness, loss of sensation, or even loss of the limb. Chronic compartment syndrome, also called exertional compartment syndrome, occurs during exercise. It's most common in athletes, particularly runners. The muscles swell during activity, and the pressure within the compartment rises to a level that restricts blood flow. This causes pain, tightness, and sometimes numbness or weakness. The symptoms resolve when the activity stops and the muscles relax. Treatment typically involves modifying activity levels, changing footwear, or in persistent cases, surgical release of the fascia. Aging brings inevitable changes to the muscular system. Perhaps the most significant is sarcopenia, the age-related loss of muscle mass and strength. Beginning as early as the third or fourth decade of life, muscle mass gradually declines, accelerating after age 60 or so. By the eighth or ninth decade, an individual may have lost 30 to 50% of their peak muscle mass. This loss affects both the number and size of muscle fibers, with type 2 fast twitch fibers being particularly vulnerable. The result is not just weakness, but also a decline in power, the ability to generate force quickly. This makes activities like climbing stairs, rising from a chair, or catching oneself during a stumble more difficult. The causes of sarcopenia are multifactorial. Hormonal changes play a role. Levels of growth hormone, testosterone, and other anabolic hormones decline with age, reducing the stimulus for muscle protein synthesis. The nervous system also changes. Motor neurons are gradually lost, leading to denervation of muscle fibers. Some of these fibers are reinnervated by sprouting from nearby neurons, but the process is imperfect, and the motor units that remain tend to be larger and less precisely controlled. Chronic low-grade inflammation, common in older adults, may also contribute by promoting protein breakdown. Lifestyle factors matter enormously. Physical inactivity accelerates muscle loss, while resistance training can slow or even partially reverse it. Studies have shown that even elderly individuals in their 80 seconds and 90 seconds can increase muscle strength and size with appropriate training. The adaptations may not be as pronounced as in younger people, but they're still substantial and functionally meaningful. Nutrition also plays a role, particularly protein intake. Older adults may need more dietary protein to stimulate muscle protein synthesis effectively, yet many consume less than younger adults. Sarcopenia has serious consequences beyond simple weakness. It's associated with an increased risk of falls, fractures, loss of independence, and mortality. Falls are a leading cause of injury and death in the elderly, and much of the risk stems from declining muscle strength and balance. Interventions that preserve muscle mass and function can have profound effects on quality of life and health outcomes in older adults. Beyond sarcopenia, aging affects other aspects of muscle function. Muscles become less flexible, partly due to changes in connective tissue. Tendons become stiffer, and the range of motion at joints may decrease. The ability to regenerate muscle tissue after injury declines, and recovery from strains or other damage takes longer. The neuromuscular junction undergoes changes as well, with some junctions becoming less efficient at transmitting signals. All of these changes contribute to the gradual decline in physical performance that characterizes aging. Yet the picture is not entirely bleak. The muscular system retains a remarkable capacity for adaptation throughout life. Regular physical activity, particularly resistance training, is one of the most effective interventions for maintaining muscle health in older age. It stimulates muscle protein synthesis, improves neuromuscular function, and helps preserve the size and number of muscle fibers. It also has benefits beyond the muscles themselves, improving bone density, cardiovascular health, metabolic function, and even cognitive performance. The principle is straightforward: use it or lose it. Muscles respond to the demands placed on them. If those demands diminish, the muscles atrophy. If the demands are maintained or increased, the muscles adapt and retain their function. This plasticity is one of the muscular system's most valuable features, and it persists even into advanced age. In the end, the muscular system is both resilient and vulnerable. It can adapt to extraordinary demands, growing stronger with training, compensating for injuries, and maintaining function across decades. But it requires maintenance. Neglect, injury, disease, or simply the passage of time can all take their toll. Understanding the ways muscles can fail and the factors that support their health provides a framework for making choices that preserve function and quality of life. Muscles are not just the engines of movement, they're metabolic organs, producing heat, consuming energy, and contributing to overall health in ways that extend far beyond their contractile function. They're involved in glucose metabolism, playing a key role in regulating blood sugar. They serve as a reservoir of amino acids that can be mobilized during illness or starvation. Their activity influences bone density, cardiovascular function, and even brain health through the release of signaling molecules called myokines. The health of the muscular system, then, is intertwined with the health of the body as a whole. This is why sarcopenia is associated not just with weakness, but with metabolic dysfunction, increased disease risk, and reduced lifespan. It's also why interventions that support muscle health, exercise, adequate protein intake, and resistance training in particular have such broad benefits. We often take muscles for granted, noticing them only when they fail or fatigue. But they are working constantly, maintaining posture, generating heat, supporting metabolism, and enabling every voluntary movement. From the first breath to the last, from the smallest gesture to the most demanding athletic performance, muscles are the biological machinery that translates intention into action. They deserve attention, care, and respect, not just for what they do, but for what they make possible. A life of movement, independence, and engagement with the world. That's no small thing. And it all depends on tissue that, for the most part, simply contracts and relaxes over and over, day after day, without complaint. At least, not until we neglect it. Then it makes its displeasure known, often when we're carrying something heavy up a flight of stairs or trying to get out of a chair after sitting too long. The muscular system has its ways of reminding us that it's still there, still working, and still very much in need of our cooperation.